the fact that we are living in a world which is occupied by a lot of microorganisms. And in fact, microorganisms, bacteria, were the first organisms on this earth. They originated roughly about 3.5 billion years ago. Humans originated much, much later. And that means we have been living throughout the human history in a world already occupied by microorganisms. And as you know, some of them, only some of them causes harm, while most of them don't do any harm. Actually, they are helpful to the this world, including the animals, including the humans. They are helpful. Why they are helpful? In many different ways. The elements in this world, for example, elements like carbon, hydrogen, oxygen, nitrogen, you know as a fact that they occur in different forms. Oxygen occur as oxides. Water is also an oxide of hydrogen. And oxygen occurs as an oxygen as the oxygen gas. So nitrogen, you know that there is ammonia, ammonium ions, nitrogen gas, nitrates, nitrites. So none of these forms of elements, different forms of elements, we can say they are different forms of elements. Nitrate is a form of nitrogen. Nitrite is also a form of nitrogen. Ammonium is also a form of nitrogen. So different forms of the same elements are there. And do you think they are going to remain as the forms, as they are? It is not. I will explain it. Ammonium ions are not going to remain as ammonium ions forever. Nitrogen gas is not going to remain as nitrogen gas forever. Nitrogen gas will be converted into ammonium ions. And ammonium ions will be converted into nitrite ions. And then nitrate ions. Then nitrate ions will be converted into nitrogen gas. And as I said, nitrogen gas will be converted into ammonia. So as you can see, there is a cycling of matter in this biosphere. And who helps the cycling of matter? Actually, all organisms more or less help the cycling of matter. You had your breakfast probably, and you are exhaling carbon dioxide. So what you had is organic matter. Now you are busy turning it into carbon dioxide. So you are also supporting the cycling of carbon. You are part of the carbon cycle. But you are not converting ammonium ions into nitrite ions, sulfide into sulfate ions. We are not doing that. And if you look at the biosphere, different organisms would contribute to the cycling of matter in different ways. And if you think about bacteria, some of them are ammonium oxidizers. They oxidize ammonium ions into nitrite ions. Some of them are nitrite oxidizers. They convert nitrite ions into nitrate ions. Some of them use nitrate ion as the electron acceptor, as their terminal electron acceptor. Don't worry too much about these words. I'm just, you don't need to understand every single word, every single science behind this, principle behind this is you don't need to understand, but just listen to me and you will understand what is the relationship between microorganisms and the world. So some organisms would convert ammonium ions into nitrite ions into nitrogen gas. So the point I wanted to make is microorganisms contribute to the biogeochemical cycle more than all the other organisms. Put together all the other organisms, microorganisms play a huge role because there is a huge diversity among microorganisms. Some of them are ammonium oxidizers while some others are nitrogen fixing organisms. So just understand that bacteria are helpful. That is the only message I wanted to give you this morning. And food microbiology, when we talk about food microbiology, automatically come to our minds that food can bring us diseases. Food may be spoiled. Foodborne diseases and waterborne diseases. B-O-R-N-E. B-O-R-N-E. Borne means carried by water. Foodborne diseases means diseases carried by infectious diseases carried by foods. So we all have heard this. Foods can bring us diseases, convey us pathogenic microorganisms. And we know how to save ourselves from them. Not only us who know microbiology. All the time in the history, people had this problem. But they survived to tell the story, as they say. We were evolved in a world where there already were microorganisms. Some of them were pathogenic. And some of them caused us diseases. Some of them killed us. But we survived. That is the story between the interaction between humans and microorganisms. Forget about humans. So many other animals are there. When they eat their foods, they eat a lot of microorganisms with them. Meat-eating animals. They kill an animal. They can't eat it in one go. They leave the carcass to come later and to eat it. By that time, it's almost rotten. Lot of flies will be there. It will be smelly. Then meat will be rotten. But they eat it with lot of bacteria. They survive. So, although there are bacteria, although they can cause us diseases, the final analysis is we survive. Animals survive. That's how this world evolved. If microorganisms can wipe us out, we would not be able to tell you the story, talk like this. We would not be able to survive. So, microorganisms are there, we are there, and now for about 100, 200 years, people know about microorganisms and people know how to save ourselves from them. And that is the knowledge accumulated in humankind for about 2 centuries. It's very developed now. Food microbiology, food preservation, how to make foods in industrial scale without contaminating them with pathogenic microorganisms and spoilage microorganisms. That's what we are going to learn today. Even without knowing that there are microorganisms, people, without knowing, they used some methods to preserve our foods. For example, Vedas, forest dwelling people, they used honey, sugar solutions, or salt to keep their food longer. They didn't know anything about microbiology. Today we are going to learn what is the basis of these preservation methods, how to prevent microorganisms entering into our foods. What are the industrial facilities, what type of measures they have to take to prevent pathogenic organisms and spoilage organisms entering into their foods. Because nowadays, early humans ate whatever left by other animals. Then they started cooking. But those days, there was not a problem of preservation was not a problem because they used to eat and there was no food manufacturing in large scale. There is no distribution in large scale. There was not keeping the foods for a long time other than, as I said, keep putting your meat in honey or a sugar solution or salt. That type of methods. Other than that, there was no need to keep foods for 2 years, 3 years in shelves. We call it shelf life. Shelf life. That does not mean that you have to keep it on a shelf in a shop. But the shelf life means, the term shelf life means after the production, before the consumption. Maybe the food is going to remain in a shop, on a shelf. There is a time that is called shelf life. How long you can keep this food without being spoiled. You know, there is an expiry date on your cake. The packet, it says expiry date. You have to eat it before this date. Date of manufacture, date of expiry. That is the shelf life. Okay. So now in the industrial scale, as we produce foods in industrial scale and distribute it, keep it before consumption for some time. So therefore, we need to know how to keep foods long, how to improve the shelf life, how to increase the shelf life. All these things are going to be discussed today. And when the industrial facility prepares a food to distribute and sell later, you have to make sure that this food is not going to be contaminated. Microorganisms have not been entered into this food. So largely excluded the presence of microorganisms. One day it will be spoiled. After expiry date, it will be spoiled. But that means few microorganisms may still be there, but largely prevented the entry of microorganisms. And there is a methodical way. Unless you know about microbiology, you can't prevent microorganisms entering into your food. You take your lunch to your workplace. You take your lunch packet. Most of you perhaps take your lunch packet to your workplace to eat it during the lunch hour. You cook that food in the morning and you serve it and you wrap it and take the lunch as a packet. to the workplace. If you fail to eat it, if you manage to eat it during the lunchtime, everything will be okay. If you are busy, if you postpone eating your lunch packet, maybe by afternoon, late afternoon, you will find that it is spoiled. You can't eat it anymore. Why? Microorganisms have grown there. Have you seen them entering into your food? No. Have you kept your lunch packet open to allow them to enter into your food? No. It has been wrapped. Microorganisms can penetrate through the paper, the tissue, and everything. They would not do that. But still, it is spoiled. So you have to understand that they must have entered into your food while you are serving it onto a lunch sheet, and before wrapping it. They might have entered. That's your guess. Otherwise, how can they grow in there? Or they must have survived the cooking process. They must have survived, some microorganisms at least must have survived during cooking. If not the vegetative cells, maybe their spores must have survived, and they germinated in the few hours time, and they grown and consumed your food, released some gases, converted food starch into some alcohol, acids. So you can't eat it. So the message I want to give you is, without knowing, without letting you know, they enter into your foods. The secret is they are everywhere. They are everywhere while you are serving, from the pan to the lunch sheet. They contaminate your food. They don't purposely enter into your food, but they accidentally fall in your food and they grow there. They are transient. They are blown in the wind all the time, everywhere. Thousands of millions of microorganisms are teeming in the atmosphere, in water, in soil, in raw foods. Everywhere microorganisms are there. And they can grow. But if you eat them before they are growing, you will be all right. So that shows us the relationship between our food and the microorganisms. Pathogens or spoilage microorganisms. They consider our foods as their foods as well. You cook for yourself, but microorganisms eat it. Let us say so. So why is that so? Macronutrients, proteins, carbohydrates, lipids, they are the main things in our food. Other than that, there are a lot of plant compounds, other vitamins, so and so. Whatever there is in your food, that's organic material. And the same organic material, organic food, is common to many different microorganisms. Proteolytic microorganisms would consume proteins in your food. Lipolytic microorganisms would consume lipids in your food. Likewise, our food, their food, no difference. We eat organic matter. There are so many bacteria which can grow upon organic matter, which can consume organic matter. Lot of different organisms, including pathogens, including spoilage organisms. So that's why the problem is there. Microorganisms and foods. We eat what they eat. They eat what we eat. So it has to, the entry of microorganisms into our foods, especially when we do it in industrial scale, we have to prevent the entry, prevent the growth of them in our foods, prevent the entry of them into our foods, which is not that difficult. If you know microbiology, you know how to prevent. The world is out there. There are so many food manufacturing plants. They prepare food and we keep them in shops. After a few months or years, we buy them and eat them and we survive. That means the industries follow methods. You can follow these methods to prevent the entry of harmful microorganisms, spoilage microorganisms, even at your kitchen. You cook and keep the lid closed. Microorganisms can't penetrate the lid and go into the food. Actually, they don't go there. They accidentally fall in there. So they wouldn't, the food would be very safe for many hours if you keep the lid closed. But you can't keep the lid closed forever. You cook the food, you open the lid to serve. That time is enough for microorganisms to enter into your food, to fall into your food. So the awareness of the fact that microorganisms are everywhere helps us. And awareness of the fact that microorganisms are everywhere helped people to develop methodical processes. You must have heard the term HACCP, hazard, and it gives you perhaps gives you a headache. But it is a simple, an intelligent way. Now you will understand it. It is not that difficult, right? It is an intelligent way how to prevent the entry of microorganisms, how to kill the microorganisms who are already entered, who have already been entered, and how to prevent the entry, how to keep the microbiological quality of your food. It's not a big deal. Intelligent way of how to do that, and we will discuss it later. First, I told you microorganisms and humans share the same food, so they grow in our food, and when we eat our food, what happens? We eat them as well. Nothing happens. We eat millions of microorganisms with water, with food. None of these things, we don't even know that microorganisms are there. But occasionally, as you know, some microorganisms causes trouble. But have you come across a person in your life? At least you are at least 20 years old, I assume, about 20. In the past 20 years, have you come across a person who, have you heard of somebody who killed by a foodborne or waterborne infection? Very rarely. But in some countries, in poor countries, so many people are getting killed by foodborne diseases. Actually, it is not the diseases. For example, in Pakistan, more than 500 children are killed every day by foodborne or waterborne diseases, I heard. I'm not quite sure about the statistics, but hundreds of children are killed. Actually, they are killed not directly by the action of the microorganism, but indirectly. That is dehydration. Microorganisms cause diarrhea, may cause diarrhea, and diarrhea can make you dehydrated. Actually, diarrhea is an efficient method of flushing out of microorganisms in the stomach, in the intestinal tract. Diarrhea is not that bad. If you don't get diarrhea, the pathogen would stay in your intestinal tract without being flushed out. But luckily, diarrhea happens. That is how evolution happened. Evolution occurred. Microorganisms cause diarrhea, but diarrhea helps us, right? We have evolved together. So this diarrhea caused by microorganisms, in a way, it serves us by flushing the microorganisms out. හේදිලා යනවා ඇතුලේ තියෙන microorganisms. But it can bring a very dangerous fatal condition, that is dehydration. Dehydration is dehydration of what? Dehydration of your blood. That is really, really dangerous. What do we mean by dehydration? Blood is an aqueous solution. There are cells, there are nutrients, there are proteins, so many things are there in blood and water. And if that water is somehow removed, the blood volume goes down. Water is needed to keep the volume. What is the need to keep the volume? There is a need. If you lose the blood volume, the heart is a pump. The heart would not be able to do the pumping action efficiently. You have to have your blood vessels filled with blood. If the volume is down, the heart cannot efficiently pump in and pump out blood. So if you cannot pump in, pump out blood, what would happen to you? The blood is the mechanism, but is the transport medium which transports oxygen. Other than nutrients and everything, it transports oxygen, which is very crucial. Oxygen to cells all over the place. You inhale air and oxygen is translocated from the environment into the blood, and the blood circulates oxygen, and oxygen is needed to produce ATP, and without ATP you cannot survive. ATP is a chemical which is needed for your biochemical reactions, and it is required in very large quantities. For example, if you sit there for 1 hour, you need 1.5 kilograms of ATP. There is no way of producing it beforehand and keeping it in storage. So the answer is to produce it non-stop. Produce it continuously in large amounts, because you use it in large amounts. So that's oxygen helps it. Without oxygen, you can't produce ATP. So without blood, you can't take oxygen to cells. Without blood volume, you can't circulate blood. Diarrhea causes the loss of blood volume, and it can bring very quick death. So that is the danger of foodborne infections. The bacterial poisons, bacterial other factors, more than everything else, the diarrhea caused by the bacterial infection can bring in death. But humans, as I said, humans survive. And we are going to discuss these things this morning. And as an organism, let us think about the need of foods. What is the need of foods for us? Actually, our body is composed of materials, structural and functional material. And we need to get the raw materials to produce our body material. from outside. That's why we eat food. If you don't eat food, you will not get any raw material to build your organic compounds. You eat organic compounds and you use those organic compounds as raw materials to build your body, to repair your body, to maintain your body. You need proteins, you need material, organic material. Your body, flesh, blood, everything is made of material. For example, red blood cells do not live more than 3 months. So you have to produce red blood cells all the time because they are dying. Likewise, you need a lot of material from outside. That's why we eat food. That's not the only reason, by the way. We oxidize our foods. Part of our food is getting incorporated into our body material. We use amino acids to synthesize proteins. You can imagine. We eat, and some of this organic matter in our food is used as raw materials to build our body material. It's not difficult to understand. Part of our food is used to build up the body. Structural and functional compounds. Proteins, cell walls, everything. Cell membranes, everything. Cytoplasm, blood cells, everything. Part of our food, actually we don't need that much of food to build our body material because you are grown up. Your body is not fully growing, but as I said, red blood cells are being produced. So, but not as much as a small child, a baby needs. Material, the baby needs a lot because it's growing rapidly. Adults, maybe not that much, but we need a lot of food for that. And most of the food an adult eat are not utilized as raw materials. What happens to them? You oxidize them. You eat carbohydrates, you eat proteins, you eat lipids, and you oxidize them. Messages keeping on popping up in my screen. I don't know how to get rid of them. Anyway. Right. I was telling you the purpose of eating foods. Part of the food is utilized as raw materials and part is oxidized. Oxidized into what? Proteins are oxidized. Part of the proteins, part of portion of the carbohydrates are oxidized fully. Lipids oxidized into what? The carbon of our organic matter. Carbon we eat. Carbon of the organic matter we eat is oxidized into carbon dioxide, and that is what comes out from your nostrils. That's what you exhale. Carbon dioxide is the oxidized product of carbon. And as I said, part of the food is utilized as raw materials to build up the body material. Part is oxidized into carbon dioxide. For what? Are you interested in supporting the carbon cycle? Indeed, you are supporting the carbon cycle, converting organic matter into carbon dioxide, but that's not what your aim means. You are oxidizing your organic foods into something else, for something else. What is the purpose? to exhale carbon dioxide? No. We do that, but that's not our aim. Our aim is to produce ATP. That's a totally different story. This is not the time to explain how ATP is produced, why ATP is needed. But all I want to say this morning is part of our food is oxidized into carbon dioxide, not to produce carbon dioxide, but to produce ATP and some other compounds. So we have to eat food. That is the idea. Why do we have to eat food? To maintain our structure, to produce ATP. So those are the twin purposes. That's why you had your breakfast. Part of that breakfast is utilized to produce your body material, and part is oxidized into carbon dioxide to produce ATP. Whatever the reason, those are the reasons to have foods. We can't avoid taking foods. We have to have foods. And as I said earlier, microorganisms are also consumers of this food. And we can't sterilize our food. We can't kill all the microorganisms and eat our food. It's theoretically possible. But how can you sterilize food? What's the point of sterilizing? You eat and you survive. What's the point of sterilizing? No need to sterilize your food. Sterilization means getting rid of all the microorganisms and their spores. Spores are heavily resistant against heat and dryness and so many things. But we can afford to have microorganisms in our food. So many microorganisms enter into our intestinal tract with foods, and only very rarely they do us any harm. So I will explain why do we have to eat these foods. There is a tunnel. Tunnel has two ends, mouth and the other end. And we put foods into this tunnel, which we call the intestinal tract. Why do we have to put the foods, insert foods into intestinal tract? Why do we have to eat them? Is there any other way to absorb them into our cells? There is no other way, and that is the way the evolution has permitted us. You eat, and then the food will be absorbed. Why is it? Have you ever thought that why the food cannot be absorbed through our skin? You keep your food in your hands. You take your food into your hands, and it's not going to absorb from there. No matter you keep it for a year, the food is not going to be absorbed through your skin into your system. You have to eat foods. Why the food is absorbed not from the skin? මගුලක් එන WhatsApp message එකක් නොකඩවා එනවා මට හොයන්න බෑ ඒක නතර කරන්න කොහොමද කියලා. Shall we close the WhatsApp? All right, let us hope for the best. So why can't the food be absorbed through the skin? Why do we have to let it absorb through the intestinal tract? What is the difference between the skin and the intestinal tract? The epithelium of the intestinal tract, the surface of the intestinal tract, including the mouth, is can you not see it? It is smooth. The cells are not rough. The cells are not, the epithelium is not rough. It does not prevent the entry of many things. It has to allow the entry of nutrients, entry of foods. The skin cannot allow. Skin is strong. It is a barrier for microorganisms. We are covered except the eyes, the mouth, the intestinal tract, and some other places in the body. The rest is covered by a strong barrier which we call the skin. Food, nutrients, many things wouldn't be able to enter through that skin because the skin acts as a barrier for entry of microorganisms, entry of chemicals, many chemicals. If you keep a pesticide on your skin, it will enter. There are some chemicals, some medicines can enter into your through the skin. You know, diclofenac. Diclofenac is a medicine, anti-inflammatory drug. And diclofenac balm, diclofenac ointment is there. And when you rub it on the skin, a portion of it is going to enter through the skin. And another example, if you keep your body open, the skin open, and apply pesticides, accidentally some pesticides can be spilled onto your skin and there is a risk. They can enter through your skin. There is some sort of passage for some chemicals, but not foods. What is the difference between the skin and the epithelium of the intestinal tract? It is soft. It can absorb matter. That is the secret. So intestinal tract has to be soft. Otherwise, how can you enter nutrients into your body? And not only that, intestinal tract is open to digestive enzymes. Digestive enzymes mostly from the liver and from the pancreas, mostly from pancreas, some from the liver. Pancreas is also located in the middle of the liver. And bile salts, that is another thing synthesized at liver. So they come through the bile duct to the intestinal tract, digestive enzymes. And without these enzymes, you wouldn't be able to break the large molecules in your food into small molecules. That is done by digestive enzymes. And without breaking them into small molecules, you can understand that they cannot be absorbed through the membrane into cells. Between the junctions of cells, between the space in between cells, that is also a passage for some food material to go in. Some other food material will be absorbed into cell, while some other food nutrients will go through the cell junctions. That means intercellular, there are no big spaces, but we call it junctions. And nutrients are absorbed through the epithelium, and none of these things are available on our skin. Enzymes are not there, absorbable cells are not there, junctions are not there, which facilitate food absorption into blood. Soft cells are there, not on the skin, but on the epithelium, intestinal epithelium. And that is why we have to eat foods. And undigested, partially digested, which the material which cannot be absorbed into our cells, our body, is going out as excretory matter. And bacteria grow in our food, and not only that, billions of bacteria grow in our intestinal tract as well. They grow in our intestinal tract, all of us. A newborn baby, only 24 hours old, bacteria started entering. By 48 hours, 72 hours, the baby's intestine will be full of different bacteria, including Escherichia coli, including lactobacillus, various lactic acid bacteria. Is it bad for us? Is it neither good nor bad for us? Or is it good for us? Actually, it is very good for us. All of us have billions of bacteria in our intestinal tract, various different bacteria. And we call it normal human intestinal microbiome. We can call it microflora. We can call it microbial populations, mixed population of bacteria. You can call it whatever you like. Microbiome is also a new term to describe the entire bacteria, entire populations, diverse groups of bacteria in our intestinal tract. And the word microbiome means something else also. That indirectly means that they are helpful. They are part of our life, part of our, not the body. They are our intestinal tract is it inside the body or is it outside the body? Intestinal tract, it is outside the body. It is just a tunnel from mouth to the anus. So it is technically, it is outside the body, although you cannot see it. It is a tube which goes through the body to the other end. It's like a canal. And it's like a tunnel. And technically it is outside the body. But you keep your food there for a few hours to allow it to be digested, allow it to be absorbed. Unwanted material, you throw it away. And bacteria play a huge role in sitting in the intestine. What are the roles they play? Some of them produce vitamins. Some of them regulate, help. Their actions will affect our immune system. Their action will affect our nervous system. People are getting more and more knowledgeable on these facts. And for microbiologists, the most important role of intestinal microorganisms, normal microbiome, is that they have already occupied the place. What is the help? What is the benefit of it? They don't allow their growth, their presence. Don't allow pathogenic microorganisms to easily establish there. They have already occupied the place. And when some organisms occupy a place, they consume all the nutrients. So pathogens would find it little difficult to establish. If they were not to be there, we could have been wiped out from the earth long time ago because of pathogenic organisms. Luckily, we have the normal flora, and that prevents the growth of pathogens in different ways. and it helps in different ways. Lactic acid bacteria, you can read not only lactic acid bacteria, many other bacteria. You can read more on this, how the intestinal microbiome helps the host, the human animal. And basically, for today's topic, you have to understand that it is an extracellular, external place, the intestinal tract is. And there are microorganisms there. And the intestinal epithelium has to be soft, not like hard as the skin, to allow nutrients to enter. And that is the weakness of it, as can't avoid. Lungs are very soft. Why? It has to allow the passage of air. But the softness allows sometimes, not all the time, some pathogens to grow there. You can have lung diseases because the epithelium is soft, but it has to be soft to absorb matter. It has to be soft to absorb matter. Eyes. It has to be soft. The tissue of the eyes has to be soft to allow light to penetrate through the cells. But it can allow, very rarely, not always, some viruses to enter. So softness is needed. Genitalia has to be soft. The epithelium has to be soft because you have to have sensations. When you touch it, you have to sense it. Much more sensitive than the rough skin. So genitalia has to be soft to have sensations. In the same time, as they are soft, as the surface is soft, some bacteria, some viruses can enter through it. Intestinal tract has to be soft to allow nutrients to come into the body system. But it may allow some pathogenic organisms to enter or to grow there at least. So soft epithelium, we can't avoid it. It is beneficial, but they are the places, soft epithelia are the places where pathogens also try to enter, but most of the time pathogens fail. If you eat your food, the absorbance starts even from the mouth. If you take something bitter to your mouth, if you take something sweet to your mouth, unless some molecules of this sweet something enter into your epithelial cells, you wouldn't feel the taste. To feel the taste, it has to enter into cells. If you keep a poison in your mouth, it enters and you will be killed. Even if you don't ingest it, swallow it, you are going to be dead. Because the poison, many poisons would enter into your system through your soft epithelium on your mouth. So from here to the other end, the epithelium is very soft. And that facilitates the absorbance. So that is the weakness also. Allow microorganisms or their poisons, their toxins to enter. So how can you prevent microorganisms growing in your food? Because what they eat, they also consume. So they grow there. How can you prevent the entry? We can start thinking about this from very basic things. If you keep your food closed, as I said earlier, they wouldn't be able to enter closed from a lid. Yes, there are small, you can't make it airtight, but microorganisms would not purposely go through a certain bendy route and go into your pan. Most of the time, if you keep the lid open, they fall there. If you keep the lid closed, they do not go through lot of barriers and turning every time and enter purposely into your food. They don't know that there are foods there. They accidentally fall there. So you can prevent them blowing in the wind, flown in the wind. They can fall into your food. So keep the lid closed. That's one thing. Eat before it getting spoiled. Cook and eat it. Or put in the refrigerator. That largely prevents, that does not kill microorganisms. Most of the microorganisms are survived at 4 degrees, no problem. They would not grow fast. Growing means microorganisms growing. A baby growing. A small baby becomes a big male human. That's not what we call growth of micro. Growth means, yes, there is a little growth. A cell maybe little smaller than a mature cell. A daughter cell, the cell divides into two. Bacterial cell divides into two and produce two cells. I'm not saying that the two cells at the very beginning are of the size of a mature cell. There is a small growth, but we don't count it. What we count as growth is the number of bacteria. If you leave one bacterium in your food, after maybe in 20 minutes, depend on the bacterium, maybe 40 minutes, maybe 35 minutes, maybe 2 days. It depends on the bacterium, but they grow means they divide and increase the population. What we mean by growth of microorganisms is the growth of the population, not the growth of the individual organism. What really matters is the growth of the population. What really matters to us is the growth of the population rather than the growth of the cell. So the growth of population, it happens in an alarming rate. Some bacteria, if all the conditions are given, they grow in 20 minutes. Every 20 minutes. Grow means divide. Increase the population by double. If two bacterial cells are there, in 20 minutes, there will be 4 bacterial cells. In 20 minutes, another 20 minutes, there will be 8. There will be 16. Exponential phase, the growth is. So, you should not keep your food until it is occupied by bacteria. Unless you really need to keep it longer, you can cook and eat it. That is one method of avoiding contaminations, spoilage, and pathogeny. Keep it closed. Eat it as soon as you cook it. Those are the methods. And you can, a fish, you bring it home. You cook it, eat it, end of the story. No involvement of microorganisms. Hardly any involvement of microorganisms. You cook it, keep it a day. You can't prevent it getting spoiled. 12 hours, maybe the maximum. After that, if you keep it open, it will be spoiled even earlier. But if you keep it closed, you may be able to keep it maybe 12 hours or so. But then it will be spoiled. What happens to the fish if you don't cook it? If you bring it home, you forget to cook it. Leave it on the table. Go to sleep. Following day, you will wake up with a very bad smell in all over the place. Why? That 12 hours period is enough for bacteria to grow on the organic matter in the fish and produce different end products, acids, alcohols, gases, you name it. So it will be very rotten and it will be very smelly. You can't cook it, you will throw it away. But what happens if you add some common salt, sodium chloride? You apply some sodium chloride on your fish. Heavily you apply sodium chloride. Do you know that if you keep on applying sodium chloride and make it dry under the sun, you know that you can keep the fish for 2 years, maybe even more than that. We call it dry fish. The same fish. Applying salt, making it little more dry, makes 12 hours into 2 years. Extend the shelf life into 2 years. Why? What is the secret behind adding salt? That's what we are going to learn now. And you must have heard Vedas, the forest dwellers, they kept their meat longer dipping it in honey. What is honey? What is salt? How can we describe it technically? What is the relevance of this salt and honey? What is the relevance? What is the exact trick behind this? How can we preserve our foods? To understand that, you have to understand the concept called concentration. Concentration. What is concentration? The knowledge of concentration, the definition. What do we mean by concentration? That is important to understand this. What is concentration, by the way? Concentration is the number of molecules. Concentration is the number of molecules present in a unit volume. Unit volume. That is the technical way of expressing concentration. Ghana meter කොච්චර තියෙනවද අණු? How many molecules are there in a cubic meter? That is the unit of the scientific unit. Not the bottle, not the pint, not the liter, but cubic meter is the unit of volume in science. So concentration is the number of molecules present in a unit volume, present in a cubic meter. So what do you think about salt and sugar solutions? What do you know about the water concentration in these environments? Environment means salt or sugar solutions. You can understand that it is full of sodium chloride. It is full of glucose. There is hardly any space for water molecules. In sugar solutions, in salt solutions, in concentrated salt solutions, concentrated sugar solutions, actually crystallized salt, there is hardly any space for water molecules. Because the entire volume, almost the entire volume, not the entire volume, almost the volume is occupied by glucose molecules or sodium chloride molecules. So how about the water concentration? The point is, the important physical feature is the water concentration is very low. Very low. Low, high. Low, High, ටිකක් තියෙනවා, ගොඩක් තියෙනවා, අඩුයි, වැඩි. These are all relevant terms. You must be able to understand. The water concentration of a salt solution is very low. The water concentration of glucose solution is very low when compared to what? මොකේට සාපේක්ෂව දාඩු. You know the cytoplasm. Cytoplasm. Every organism is made of cells. And every cell has a cytoplasm inside. And there is some water there. Why do we need water in a cell? It acts as a reactant. Water is a reactant to many reactions. Water is a reactant to many reactions. Is that the only service? No, no. Water serves as the medium for all the reactions. You must have heard this, but you may be interested to know the exact meaning of how the medium serves a reaction. You know, in one place of the cell, there is an enzyme. In one place of the cell, there is a reactant. In another place of the cell, there is another reactant. Can you imagine the enzyme, the two reactants would ever come together unless there is water for the reactants and the enzymes to float? කවදාවත් වතුර නැත්තම් අර reactants දෙක දෙතැනක තියෙයි. enzyme එක තව තැනක තියෙයි. They are not going to come across each other for the reaction, and water is the medium which helps them to float in water and accidentally come across reactants, enzymes. They float. Unless you have water in the cell, none of the things would be able to meet each other. Reactions would not happen. So water is needed as a reactant. Not only that, water is needed as the medium. So you can understand that the cell has to have some water in it. Otherwise, the cell is going... See, I told you dehydration can kill a person. Inside the cell, there has to be water. The blood has to have the blood volume. That is the immediate reason for the death. In addition to that, the cells also need water. Water is a reactant. And not only that, water is the medium. But inside the cell, how many other things are there other than water? You would say salts. Yes, salts are there. Is salts the only thing in your cell? Enzymes, other proteins, ribosomes, DNA, RNA, mitochondria. Are they not inside your cell? Are they not occupying the volume of the cell? They are. Waste material before throwing away. Nutrients before being utilized. All the enzymes. So the cytoplasm is a thick solution, very thick. So many things are there. Proteins, DNA, RNA, you name it. And so is there a big space for water? No. The water concentration of the cell is also not very high, but higher than the water concentration of a salt solution, higher than the water concentration of a honey solution, because it is even thicker than the cytoplasm. So how can it prevent the microbial growth? How can it make the cells dry? Under normal circumstances, as I already explained, cytoplasm is full of different material. Some water is also there. Outside the environment, can you expect honey or salt all the time? No. Most of the time, what is the outside environment? What is the water concentration of the outside environment of a cell? Under normal circumstances, the water concentration is higher in the environment outside the cell. Under normal circumstances, the water concentration is high because there are no very many other solutes in that water in the outside the environment. Inside, the water concentration is low. Outside, the water concentration is high. Water flows. Now listen. Water, we don't need to drag water into the cells. Water automatically flows into cells through protein channels in the cytoplasmic membrane. Water diffuses into the cell along the concentration gradient of water. As I said, under normal circumstances, inside the cell, the water concentration is lower than that of the outside environment. That means water concentration of the outside environment is higher. Water concentration under normal circumstances. Under normal circumstances, water concentration generally in the outside environment is higher than that of the inside environment, inside cytoplasm. So water goes along the concentration gradient into the cell. And the Vedda comes and apply honey to the environment. The fisherman, dry fish maker comes and apply salt. What would happen to the water concentration in the outside the environment? Now it is lower than the water concentration of the cytoplasm. So what happens? Water goes out. If you apply salt or sugar, water comes out from meat cells. Water comes out from fish cells. And the fish becomes dry, dehydrated. The meat becomes dry, dehydrated. Why? You have applied salt or sugar. The concentration of water is low, make it lower than the cytoplasm and water comes out. How about bacteria? If a bacterial cell faces this environment, the same thing would happen to it. What would happen? Water comes out. If you put a bacterium in salt, the water comes out from the cytoplasm into the salt. If you put a bacterium in a honey solution, glucose solution, water comes from the bacterial cytoplasm outside. That means in the death of the bacteria. They cannot grow. So, in the normal fish, the outside environment, water goes into the fish cells, the water goes into bacterial cells. When you apply salt, sugar, water comes out from the fish cells, water comes out from the bacterial cells, the fish becomes dry. Unless there is enough water, bacteria fail to grow there. And not only that, the bacterium will be dead by dehydration. That is one method of food preservation. People, without knowing anything about microbiology, they started doing this for, maybe thousands of years. They have been doing this. That is another method of food preservation. Keep it closed, eat it before being spoiled, put it in honey or sugar or salt. Any questions? Are you there? I can't feel that students are listening to this. It's very difficult for me to speak to a computer screen unless you show that you are alive and listening to this. Can you understand this? Kanishka or Kanishka? APGS Pereira, Nevindi, Himasha. Unless you say something, at least say good morning. I am not interested in doing this, continuing this class. Can you not speak? I called few of you by names. You chose to remain silent. What would happen if I chose to remain silent? Can you not speak? That makes me very angry, you know. Good morning. Okay, I will stop this class for few minutes until you learn how to speak. So that is one few methods of food preservation. And cooking is another method of preservation. Cooking, you can keep it little longer. Cooking make it softer also. If you know lentils, dal, you can keep them dry dal maybe few years. But if you cook them only few hours, the dal curry will be spoiled because the material will absorb lot of water and partially degraded due to heat, so easily spoiled after cooking. Vegetables, they will be spoiled after cooking, but in the same time you have to understand that cooking at 100, nearly 100 degrees kill lot of vegetative cells. You can't say no to that. It kills lot of bacteria. On the one hand, it helps the bacterial growth. On the other hand, cooking kills bacteria, and cooking is one such method. And not allowing oxygen to enter into your food, that is another method of preservation. Oxygen. What is the role of oxygen? Oxygen, in the beginning of this world, there was no oxygen gas. Slowly, oxygen started accumulating in this biosphere, and some organisms were unable to live in the presence of oxygen. They confined to anaerobic areas, while some organisms learned how to face oxygen. These are not really food microbiology, but I have to mention, otherwise how could you understand this? Oxygen is a chemical. Oxygen can remove electrons from many biochemicals. That means oxygen is a strong oxidizing agent. And we need it also, but we don't allow oxygen to be free inside our cytoplasms. Instead, we take oxygen to the electron transport chain of our mitochondria. We transport oxygen in a bound form. Oxygen is not free to react. Oxygen is bound to hemoglobin. And oxygen is released only at the end of the electron transport chain. And oxygen is not allowed to be free in the cytoplasm. If it is allowed to be free, it would react with many biochemicals, starting even worse. Oxidation of biochemical is bad, but the oxidative products of oxygen are even worse than oxygen. I very briefly mentioned, if you have questions, you ask from me. If you ask, I will explain. If you don't ask, I would not like to spend too much time on these related facts. They are related, but it takes time. This workshop has only, 1 day, and I have to finish many things. You have paid for this. So I can't spend too much time discussing some not so directly relevant things. But still, if you ask me a question, I am ready to answer. Willingly, I would answer. So oxygen is needed for aerobic respiration, but free oxygen in cells is not favorable. What happens is they remove electrons from many biochemicals. That itself is bad. And when they remove one electron from a biomolecule, when oxygen molecule O2 removes one electron from a biomolecule, it produces a very strong oxidizing agent, much more stronger than oxygen itself. It is called superoxide anion. Superoxide anion, O2, that is a dot on the top and a minus mark, O2 dot minus. That is superoxide anion. That is a very strong oxidizing agent, means that removes electrons from many more biochemicals. That is bad first. So we have mechanisms to destroy these superoxide anions. And it produces hydroxyl anion. We have enzymes to destroy hydroxyl anions as well. We have peroxidase, we have catalase, we have superoxide dismutase. In short, what we have to fight against the toxic forms of oxygen is antioxidant mechanisms. You are asked to eat fresh vegetables, fruits, and they also contain antioxidants, vitamins. Some of the vitamins, some phenolic compounds in plants act as antioxidants. Even if you don't eat anything antioxidant, any antioxidant, you still survive due to the inbuilt antioxidant mechanisms in your body. If you eat antioxidants, it supports you to fight against reactive oxygen species. I am very briefly going through it. But the point I wanted to make is, earlier there was no oxygen in this world. Slowly oxygen appeared due to photosynthesis of some organisms. Oxygen became very bad to organisms. And some organisms as a result are confined to anaerobic areas where oxygen is not there. And those who live in the presence of oxygen have mechanisms to face against toxicity of oxygen. Not only that, we learn how to manipulate oxygen in aerobic respiration. If you have questions, you have spent money on this course, you can ask. If you have questions and keep your mouth shut, you are not going to learn these things in your life. That's my guess. And I am willing to help you, but how can I help you without you play dumb? DUMB dumb. කතා කරන්න බෑ ගොළු. හරි? It serves you. If it serves you better, you stay silent. කතා කරන්න එපා එකෙක්වත්. Good morning කියන්නවත් එපා. Good afternoon කියන්නවත් එපා. You remain silent, and it's not going to do me any other than me getting angry. It does not do me any harm. It harms you. If you have questions, anytime you can ask, and you are always welcomed. Ravindu, are you listening?
Yes,
sir. Next time when you have to
please unmute and say something. Yes.
Yeah. It's very difficult for me to talk to a screen without knowing that none of the... Can I try it again? Ravindu, would you permit me to name them and ask them at least to say good morning? Kanishka?
Yes, sir. Yes, sir. It's fine.
Goluvik. Netmi? Goluvik.
Morning, sir.
Good morning. මං උදේ ඉඳලා කතා කරන ඔයාට මොකද කතා කරන්න බැරි? Netmi, from where you are? කොහෙද ගම? කොහෙද ඉන්නේ? ගෙවල් කොහෙද තියෙන්නේ? අභිරහසත් දෙක කියන්න බැරිද?
I'm from Piniandala, sir.
Sorry?
I'm from Piniandala.
You are from Piliandala. Good morning to you.
Good morning, sir.
Do you have any questions?
No, sir. You have described things really well.
Oh, thank you very much. I'm glad to hear that. I'm glad to hear that. My frustration is over, vanished. මම නිකන් තරහ ගිහිල්ලා හිටියේ. frustration මම වයසක මිනිහෙන්නේ. frustration කියන එකේ තේරුම දන්නවද නෙත්මි?
I can understand.
නොසන්සුන් භාවයක්. හරි. And getting angry, getting frustrated. So now I am happy. Thanks for making me happy. Right? So if you manage to understand that, that makes me happy. If I feel like some of you could not understand, I have to explain it again. The only reason I am asking whether you manage to understand is, what do you think? I ask this question because then I can explain it again. If you cannot understand, I can explain it again. That's my job, you know. So anyway, so මොකද්ද මම අන්තිමට කිය හිටියේ නැත්මි? Antioxidants, right? Antioxidants. So you have heard the story of good morning. Thank you. Dulen කොහේ ඉඳලා කතා කරන්නේ? දුලන්ද අමාර අය බයයි මේ අන්මියුට් කරන්න මේ එන්න විශාල හදයක් ඇහෙන හින්දා. From නුගේගොඩ. Okay, thank you. ගෙවල් වල ගොඩක් මරා ගන්නවා නම් එහෙම ලොකු සද්ද නම් අන්මියුට් කරපුවම ඒව ඇහෙන හින්දාද කොහෙද සමහර ළමයි කතා නොකරන ඔක්කොටම මම කියන්නේ ඒක. හරි. ඕගොල්ලෝ in a very difficult situation because your households, maybe so many people are using bad words, fighting, killing each other, and that's why you can't, I'm insulting you. That's why you can't unmute yourself. එහෙම කිව්වහම මගේ තරහ ටිකක් අඩු වෙනවා. ඕගොල්ලන්ගේ ගෙවල් වල මරා ගන්නවා ඇති ඒවා අපිට ඇහෙයි කියලා තමයි unmute කරන්න බය. කියලා කියමුකෝ අපි. සමහර අයගේ ස්පීකර් වැඩ කරන්නේ නැතුව ඇති, මයික්‍රොෆෝන් වැඩ කරන්නේ නැතුව ඇති. ඒවත් ඕකේ. සමහර අය බයයි මේ ප්‍රශ්නයක් අහන්න. තේරුණා කියන්නවත් බයයි, good morning කියන්නත් බයයි. ඇයි මොකද එහෙම වෙන්නේ? අපි ඔක්කොම මේ රටේ මිනිස්සුනේ. මම ඔයාලට උදව්වක් කරන්නනේ ඇවිල්ලා ඉන්නේ නේ. මගෙන් ප්‍රයෝජන ගන්න මේ වෙලාවේ. මං ප්‍රශ්න ඇහුවට තරහ වෙන්නේ නැහැ යාළු වෙනවා මේක තේරෙනවනේ දැන්. Anyway, so antioxidant story came and I have tried to explain it very briefly because it might be important for you. Antioxidant කියන જાතිය දැනගන්න නැතුව වැඩක් නැහැ නේ ඉඳලා. එහෙම දෙයක් ලෝකේ තියෙනවයි කියලා. So the main thing I wanted to talk about is oxygen. The role of oxygen. Oxygen is important for us for aerobic respiration. Aerobic respiration converts organic matter into carbon dioxide. And that is the fastest method of degrading organic matter. We have fermentation. In this biosphere, there is a process called fermentation that also oxidizes organic matter only partially. Glucose into acetic acid. Sucrose into acetic acid. Starch into acetic acid. Starch into butyric acid. Starch into acetic acid. Starch into lactic acid. So and so, that is ethyl alcohol. Sugar into ethyl alcohol. That is fermentation. Degradation, but partially. Oxidation, yes, but partially. Aerobic respiration. Aerobic respiration oxidizes organic matter completely. So, if you allow aerobic respiration to happen in your food, is that not the fastest method of degradation of your food? That is the fastest method. If you allow aerobic respiratory bacteria to grow on your food will be spoiled very easily, very rapidly. If you allow if you cut off oxygen supply, I am talking about cutting off oxygen supply is another way of preserving food. Preserving food means not allowing food to be degraded microbially. So aerobic respiratory microorganisms, the growth of aerobic respiratory microorganisms, you can stop it completely by doing what? Threatening the microorganisms, giving poison to them? No, you can simply cut off the oxygen supply. You can keep the food anaerobic. You can keep it closed. You can make it vacuum packed. You can suck the air out. So by doing so, you can make the food without oxygen. Dissolved oxygen is finished. That way, what can you stop? You can stop the growth of microorganisms. All microorganisms? No. Aerobic respiratory microorganisms. Yes. What's the point of stopping aerobic microorganisms, growth of them in your food? Those are the organisms which oxidize the food, degrade the food most rapidly. So better than nothing. Stopping the growth of aerobic microorganisms help to prevent the growth of aerobic microorganisms. So that is also preservation. ගොඩක් microorganisms ලා නතර කරගන්න පුළුවන්. How about fermentation? Is it allowed to happen? Is it possible to happen in an oxygen-free environment? Yes. Yes. But that happens if you leave a lot of microorganisms in your food. If you cook it, if you sterilize it, if you autoclave it, if you put it in the pressure cooker, these methods will reduce the microorganisms to a great extent. Autoclaving would kill all the microorganisms, including their spores. Cooking, some vegetative cells, not all, not spores. Spores cannot be killed by cooking. But the load of microorganisms, the number of microorganisms too, can be reduced to a great extent by cooking, by pressure cooking even more, by autoclaving, complete eradication of microorganisms. How about cooking and cutting off oxygen supply? Cooking plus cutting off oxygen supply. You stop the growth of aerobic respiratory microorganisms. Cooking reduced the number of microorganisms to a large extent. So you may be able to keep the food little longer. You autoclave the food, kill all the spores, and kill all the vegetative cells, and keep it oxygen free. Keep it in a can or a vacuum pack. That way you will be able to keep the food much longer. You pressure cook it, the food, and then vacuum it. Pressure cooking would kill a lot of bacteria. Vacuuming, making it a vacuum would cut off oxygen supply. Putting it in a can would cut oxygen supply. So aerobic respiratory bacteria will be stopped, and the microbial load also will be reduced to a great extent, so you can keep the food longer. Think about aerobic respiration. Think about providing oxygen. Think about providing oxygen. That way you can achieve the degradation of organic matter very quickly. If your aim is to degrade organic matter, in food microbiology, we want to keep the food longer. But maybe wastewater treatment, you want to make the organic matter degraded as early as possible, as rapidly as possible. So what do you have to do? You have to provide aerobic conditions. You have to aerate it. In wastewater plants, wastewater treatment, do you know something called wastewater treatment? Wastewater treatment means getting rid of organic matter in your industrial wastewater, getting rid of organic matter in your industrial wastewater before the water is released to the atmosphere, to the environment, So you allow aerobic respiratory bacteria to do the job for you. Wastewater treatment, organic matter, complete oxidation of organic matter into carbon dioxide. So how to do that? You have to aerate it. You have to provide oxygen to the wastewater. So wastewater, when you oxygenate it, the bacteria would oxidize it very quickly, very rapidly. Can I give you another example to help you to understand this aerobic respiratory bacteria, the growth of aerobic respiratory bacteria? The growth of aerobic respiratory bacteria would degrade your food, degrade the organic matter very rapidly. Can I give you another example? Take a forest flow, the flow of a forest. There is a forest, the ground. There are a lot of organic matter in there. Organic matter, maybe a very thick layer of organic matter, perhaps even 1 meter thick, as 1 meter high organic matter is there on the forest floor. Dead animals, dead plant matter, excretory matter of organisms, dead microorganisms, dead worms, you name it. The forest floor is teeming with organic matter, very rich with organic matter, and it's being slowly degraded. The organic matter is not going to remain there. අලියෙක් මරුණාම අලියා විදිහට ඉන්නේ නැහැ නේ, ඒක දිරනවා. So the organic matter on the forest floor is subjected to degradation. And then the rain comes. What would happen to the organic matter? Some of it is carried by water to the streams, to the rivers, which are flowing across the forest. Have you been to a forest? Have you been to Sinharaj? Have you been to these wilderness areas? Okay, Arin. නෑ ඒකට කමන්නේ. අරින්නකො. අපි කරුවල. දාඩිය දාන්න. So have you seen streams, water flows running through a forest? Are they very dirty, smelly, opaque, or very transparent, crystal clear? Very clean water you can see. What happened to the organic matter? It kept on flowing into the rivers, into the waterways. But as long as the water runs very rapidly over logs, over stone or rocks and logs, downhill very fast. When the water flows very fast, oxygen getting mixed with water, the water is aerated. That's why there is hardly any organic matter in that water. What happened to this organic matter? Has it reached the stream? Yes. Water flows downwards. Water coming from the forest floor. Imbibed, lot of water is imbibed in the forest floor and slowly coming to the river. And when the rain comes, a lot of water comes to the river. But still the river looks very clean. Actually, it is very clean. It is crystal clear. Why? Aerobic respiration, because the water is rapidly oxygenated, and aerobic respiration degrades all organic matter. And you can see the bottom of the river. Very clean. But the water, if the water is stagnated, if the water is not moving fast, so no oxygen is dissolved, Therefore, organic matter cannot be degraded by aerobic respiration. So organic matter would remain in the water. So you wouldn't see clear water if the water is stagnant. If it flows, oxygenated, and organic matter will be degraded. So what is the relevance of this example to food microbiology? If you allow oxygen, the food will be degraded rapidly. So if you cut off the oxygen supply, it stops the degradation of food by aerobic respiration, the most rapid way of organic matter degradation. So keeping oxygen away from your food you would be able to keep the food longer. How about fermentation? I asked the question earlier also. Fermentation can happen, but if you cook it before packing, the number of microorganisms will be reduced, so no very much possibility for fermentation as well. How about anaerobic respiration? Have you heard something called anaerobic respiration? What is the difference between aerobic respiration and anaerobic respiration? Anaerobic respiration is exactly the same thing, same mechanism of aerobic respiration. The only difference is the terminal electron acceptor is not oxygen. The terminal electron acceptor could be something but not oxygen. Could be nitrate, could be ferric, could be sulfate, could be anything. Hundreds of different inorganic non-oxygen electron acceptors are there. We call it anaerobic respiration. But your food, even if you cut off the oxygen supply, yes, I said fermentation can happen, but you reduce the microorganisms before vacuum packing it. So that way you can keep it longer. How about anaerobic respiration? As you may be able to understand by now, for anaerobic respiration to happen, you have to have nitrates or sulfates or other terminal electron acceptors in your food. And there is no guarantee to say, no way to say that the food is teeming with these electron acceptors. ගොඩක් වෙලාවට ආහාරවල anaerobic respiratory electron acceptors ලා නැහැ. ඉන්නවා වුණත් අඩුවෙන් තියෙන්නේ. So there is no high possibility for anaerobic respiration. So vacuum packing helps you to keep the food little longer. How about radiation? Gamma radiation. UV radiation. UV radiation can kill bacteria, but UV cannot penetrate through even paper. You know, newspaper, paper. කඩදාසි කියන්නේ. UV would not be able to penetrate through even paper. So UV is not a very efficient method of food sterilization to keep bacteria, to kill bacteria in foods. But gamma radiation, ionizing radiation we call it. It makes the elements into ions, ionizing radiation. Elements into ions, turns elements into ions means destroying all the biomolecules, inorganic chemicals. So ionizing radiation can kill bacteria for sure. It destroys DNA, destroys proteins, destroys lipids. So ionizing radiation is a method of preservation, a very good method that matter. It does not change the cooking, can change the taste of the food, but ionizing radiation may not change the taste of the food that much. So that kills all the bacteria. That is a good method of bacteria and their spores. Ionizing radiation. Refrigeration can reduce the growth, but cannot kill them. I mentioned this also earlier also. Refrigeration cannot kill bacteria, but slow down their growth. So you experience this, you know it by experience. Refrigeration can keep your food little longer, few days more, but not forever. Freezing can keep it little longer. Freezing, minus 18 degrees is the temperature of deep freezers. Freezing would stop the metabolic activities of microorganisms. In that frozen, it cannot kill microorganisms, by the way. It can make the microorganisms dormant. That means without doing any metabolic activity. So they are frozen. Their mechanisms, biological mechanisms are stopped, but not eliminated, but stopped. And they will be reactivated again once the temperature is normal. So freezing is one method of keeping food longer. You know, in your freezer, you can keep meat, fish a very long time because microorganisms are unable to grow there. They are dormant. Their growth is suddenly stopped. That's all. So freezing is another method. Autoclaving is another method that is strong enough to kill bacterial spores also, not only vegetative cells. High temperature would kill vegetative cells. Autoclaving, keeping the pressure, allowing the pressure to be increased. Inside an autoclave, the lid is closed, the valve is closed. Unless the pressure is increased up to a certain level, the valve remains closed. So the water remains pressurized and heat comes. And under pressure, the boiling temperature of water is go up, elevated. So in the autoclave, you can get a very high temperature, and the water vapor. If you keep the water boiling in a pan, open pan, what is the maximum temperature you can get? At mean sea level, the maximum temperature you can get is 100 degrees. If you go up a hill, the pressure is low, so the water will be boiled little lower than 100 degrees. If you keep the pressure on, if you don't allow the water vapor to escape, that means you are applying pressure in a closed vessel, applying heat. The boiling temperature is elevated, and 15 pounds per square inch, that's an old unit. That is the pressure of an autoclave, 15 pounds per square inch. That is how the valve is made. That is the valve is adjusted. වර්ග අඟලට රාත්තල් 15ක පීඩනයක් එනකල් valve එක වැහිලා තියෙනවා. ඊට වැඩි වුණාම පීඩනය ඇරෙනවා. That is the mechanism of the autoclave. It remains, the valve remains closed until the pressure reaches 15 pounds per square inch, and then it opens. And when the pressure is released, the valve is closed again and allowing the water to continuously pressurized at 15 degrees per square inch. 15 pounds per රාත්තල් කියලා පරණ එකකක් තියෙන 15 pounds per square inch. ඒක තමයි මන් දන්නේ ඒකකේ. And that at that pressure, what is the temperature of water you can get? The boiling temperature. Boiling temperature happen to be 121 degrees. අර pressure එක apply කරලා බලපුවම 121 තමයි වතුරේ උෂ්ණත්වය. දැන් හිමාලයේ උෂ්ණත්වයත් මේකයි 121. Unless you keep the lid closed, you cannot get water vapor hotter than 100 degrees. But now in the autoclave or even in the pressure cooker, you can get water vapor exceeding 100 degrees. In the autoclave, the valve is set to be opened at 15 pounds per square inch. That means the temperature of the water inside the autoclave is 121 degrees. That is the temperature of the water vapor as well. And that temperature, that 121 degrees water vapor can penetrate into the bacterial spores and killing the bacterial spores as well. That is the reason for autoclaving. That's the success of autoclaving. Kill all the spores and the vegetative cells. There is something called pasteurization. Another preservation method, pasteurization. Milk is going to be pasteurized. Milk will be pasteurized. Fresh milk කියලා තියෙන්නේ ඇත්තටම fresh milk කියලා කියන්න ඕනේ fresh milk කියලා ඇත්තටම කියනවා නම් හරකගෙන් ගත්ත කිරි වලට තමයි ඇත්තටම fresh milk කියන්නේ. සම්පූර්ණ නැවුම් කිරි. මේ දැන් හරකගෙන් ගත්තාම එළියට that is fresh milk. And that may contain microorganisms coming from the skin of the cow. So it can be contaminated a bit. And you can pasteurize it. Still you call it fresh milk. Pasteurize it. What is pasteurization? Who is named after the great scientist called Louis Pasteur, a French scientist. Louis Pasteur, he found so many important things. He discovered so many things which are immensely helpful to mankind. And he found this method as well. He invented an exposure to heat, not 100 degrees, but lower than that, maybe 85 degrees, 80 degrees, 90 degrees, something like that for a very brief period, 10 seconds, 30 seconds, 20 seconds. It depends on the size of the vessel. කිරි දාන භාජනයේ ප්‍රමාණය අනුව pasteurization ඔයා heat කරන විදිහ අනුව there are different time temperature combinations. There is no fixed time temperature combinations. Sub boiling point, lesser than 100 degrees. That is the temperature, maybe 85, something like that. Time less than 30 seconds. So that way, get rid of is enough to get rid of most of this spoilage bacteria and tuberculosis. Mycobacterium tuberculosis will also be killed under those conditions. And Pasteur realized that milk can be kept a little longer and the risk of tuberculosis is also lowered a lot by pasteurization. So people have been practicing this for ages. That is pasteurized milk. We can still call it fresh milk. And then ultra high treatment. Ultra high treatment. Ultra heat treatment. That means very high heat, ultra heat treatment, UHT. Ultra heat, not just ordinary heat, ultra heat. Very high temperature, but to keep the taste of the milk intact, that is also not done for hours. Very high temperature, killing a lot of bacteria, and you can keep UHT milk for much longer. You don't need to refrigerate it. You can keep it on the shelf. And although people call it fresh milk, although they are labeled as fresh milk, maybe in small letters they say made from fresh milk. So you don't need to call it fresh milk. Although the container says fresh milk, it's not fresh. UHT milk, pasteurized milk. fresh milk. එහෙම තමයි හරිම ක්‍රමය. හරකා ගාවින් ගන්න කිරි fresh milk. Pasteurized කරපුවම pasteurized milk. You can, if you want, you can call it fresh milk. UHT milk, it's not very fair to call it fresh milk. You can call it UHT milk, sterilized milk. You can keep them maybe 6 months, even more than that. So that is heat treatment. Filtration is another method, but filter, you cannot use any filter to get rid of bacteria. You have to use microbiological filters. That means the pore size is very small, 0.45 micrometers. 0.45 micrometers. Or 0.25 micrometers. But the thing is, can you filter your food? Filtration is not a good method because your food is very thick or solid. Even if it is a liquid, it is very thick. So you may not be able to filter them. You cannot use the oven also, hot air oven. That would dehydrate your food in a way that you wouldn't be able to eat it anymore. Completely dry. බත් ටිකක් අරගෙන දැම්මොත් හාල් වාගේ වෙලා තමයි නතර වෙන්නේ. Completely dry. You wouldn't be able to eat it. It's not going to be soft. So ovening is also not a good method of sterilizing foods. How about adding chemicals? Lead, mercury, heavy metal ions. They can kill bacteria, of course. But can we add these into foods? No chance. Because those things can kill us as well. They are cytotoxic, toxic to humans as well. We call it cytotoxic. Antibacterial, bactericidal, but in the same time cytotoxic. Cyto means in here, in this word, what we mean by cytotoxic is toxic to our cells. Antibacterial means bad for bacteria. So you can't add these chemicals, but there are few chemicals you can add to your food. No problem. What are these chemicals? Different chemicals are there. Thank you. Where? That's good. Thanks. Thank you. Right. I will drink it. මොදලද හරින් ගිහිල්ලා පැත්තේ හදාගෙන බොන්න. Has made some tea for me. I will drink it. She can't speak. My daughter can't speak, but she is a very nice person, but she thinks that I am here sitting here for a long time. So she has made a cup of tea and brought it to me. Thank you. What good hadagen? Anyway, sorry about the brief disturbance. අද මේ මැසේජ් එවලා තියෙන්නේ බලමු. I cannot even see the message. Good morning, sir. I understood everything so far from Muge. Good. Oh, thank you. Right. Thanks, Dulan, for sending me that message. Anyway, chemicals, adding chemicals. Obviously, poisonous chemicals would kill bacteria, but for obvious reasons, we cannot add these chemicals to foods. But there are some chemicals we can add. Weak acids we can add. Antioxidants we can add. Remove oxygen in our food. We can add these things. And I give it to you because this information is available as the time runs very fast. I have to teach you many things. So I will give you the food preservative chemicals, the chemicals which can be added as preservatives to food. And there are a lot of information about these things in the internet or in books. You can read and find out, gather the information by yourselves. I would like to explain things which are not easily found in, not easily available, not readily available in the internet and in many other resources. How about addition of antibiotics? Our topic is how to get rid of bacteria in our foods. Antibiotics are strong antibacterial agents, you know that. Why can't we add antibiotics to our foods? We can add, but we are not allowed. We are not supposed to do so. What is the reason? You know, you must have heard of the reason. What is the reason? They are worried about the emergence of resistant varieties of pathogens. Resistant varieties. Resist to what? Resist to antibiotics. You know, I take amoxicillin and it kills my pathogens. If I keep adding amoxicillin to my food, can it kill bacteria in my food? Yes, of course. But I am not supposed to add amoxicillin as a food preservative to my food. Why? As I said, they say it allows the resistant bacteria to emerge. Do you know the exact mechanism how it happens? Antibiotics දාපුහම antibiotics වලට ඔරොත්තු දෙන බැක්ටීරියා එතන හැදෙන්නයි antibiotics නොදා හිටපුවම ඒව හැදෙන්නේ නැහැ කියන්නයි මොකද්ද පදනම antibiotics වලට බැක්ටීරියා මැරෙනවා. What do you think? Any idea? Do you know? Can you give me yes or no answers? Yes කියන්න හරි no කියන්න හරි අමාරු නෑනේ. Antibiotics වලට බැක්ටීරියා මැරෙනවද?
Yes, sir.
Thank you. එතකොට antibiotics food වලට දැම්මොත් ඒකේ තියෙන බැක්ටීරියා මැරෙනවද? ඒකටත් උත්තරේ yes. ඉතින් antibacteria අපේ ඇඟේ ඉන්න බැක්ටීරියා මැරෙනවා නම් ආහාරයට කෙලින්ම antibiotics දාපුවම බැක්ටීරියා මැරෙන්නේ නැද්ද? මැරෙනවා. But we are not supposed to add antibiotics to our food. Why? Because they are worried about emergence of resistant bacteria. Can I take few minutes to explain the mechanism of emergence of resistant varieties and the relationship that to antibiotics? I will explain. If you add antibiotics in enough concentration, it can kill bacteria, no problem. That's why you are advised to have 500 milligrams of amoxicillin every 8 hours for 3 days. Every 8 hours for 3 days, you have to take 500 milligrams of amoxicillin. That keeps enough amoxicillin concentration in your blood for 3 days to wipe out the pathogen population entered into your body. Amoxicillin milligram 500ක් පැය 8කට සැරයක් පැය 8කට සැරයක් දවස් 3ක් තිස්සේ බිව්වොත් ඇඟේ ඇමොක්සිසිලින් කොන්සන්ට්‍රේෂන් එකක් තියෙනවා ලේවල ඔයාගේ ඇඟට ඇතුල් වෙච්ච බැක්ටීරියා ඔක්කොම සම්පූර්ණයෙන් මරලා දාන්න. ඔයාගේ ආහාර වලට ඇමොක්සිසිලින් කඩලා දැම්මොත් are you sure about the concentration? You don't know about the concentration. It could be higher, then there will be no problem. All the bacteria will be killed, but the problem starts when the concentration is lower. There is a little antibiotic, but not enough to kill bacteria. And bacteria keep on dividing. Reproduction means the copying of DNA. Bacteria reproduce very fast. I told you earlier also, every 20 minutes bacterial cells, not all, E. coli divides in every 20 minutes. So bacterial reproduction rate is very fast. Growth rate is very fast. That means the DNA is copied very fast. If a human reproduction, DNA is copied once in a while a baby is born. So the DNA is copied and the copies go to the new baby only very rarely, once in a while. Bacteria are not like that. Every 20 minutes, if the conditions are good, E. coli divides in every 20 minutes. So can you imagine the rate of copying of DNA? If 1,000 bacteria are there, in the next minute, in the next 20 minutes, 1,000 new DNA copies will be formed. If billion bacteria are there, in the next half an hour, there will be another billion DNA copies. So the more you do the copies, the chances of mutations becomes little higher. DNA copying is the basis of reproduction. And every time when DNA is copied, an exact copy is produced. පොඩ්ඩක්වත් වෙනස් නැහැ. DNA sequence එක, nucleotide sequence එක the same in the new DNA. But when it happens over and over again, there is a chance for some errors in DNA copy. We call them mutations. And there are error correction mechanisms also. And some mutations are strong enough not to be corrected by error correction mechanisms. And these mutations can vary a lot. It could be anything. Most of the mutations, are they good for the bacterium or bad for the bacterium? If the daughter cell is different from the mother cell, parent cell, if the daughter cells are different from the parent cell, is it good or is it bad? Most of the time, a large number of times it's bad because the bacterium is in a certain genome. It has to have a certain genome to survive. That's how it survives like the parental cell. But if the DNA is changed, most of the time it is bad, detrimental. The cell would not be able to survive. The daughter cells would not be able to survive. But occasionally, very randomly, not purposely, but randomly, occasionally, randomly, there is a possibility of mutation occurs, and that mutation makes the bacterium capable of producing, altering, already producing enzyme. That's what most of the time happens. Changing an already existing enzyme into an into an enzyme which can destroy the antibiotic. This can happen very rarely. Can you understand this? Bacteria divide. Bacteria grow in your food. You add antibiotics to food. I told you it is bad. Now I am explaining why is it bad. If you take antibiotics in the prescribed dose, prescribed duration, that's going to help you. That kills bacteria. In the food, you may exceed the prescribed concentration, required concentration, or it's okay. If you exceed the required concentration, that's enough. That's all right. Killing the bacteria, why not? But if the concentration is not enough to kill the bacteria, it's sublethal concentration of antibiotic. Then bacteria keep on dividing. Accidentally, there is a possibility of emergence of a resistant organism, meaning enzyme or something, an enzyme or some other factor which acts against the antibiotic. So this organism has a chemical, has a new enzyme to face the antibiotic. Only that organism. All the other normal wild type, we call it wild type, parental type. All the other bacterial cells would not be able to survive because there is a concentration of antibiotic in the environment. But the resistant organism can grow. That means we inadvertent කියන වචනේ දන්නවද? අපි බලාපොරොත්තු වෙන ඉෆෙක්ට් එක නෙවෙයි. අපි බලාපොරොත්තු වෙන්නේ ආහාර වලට බැක්ටීරියා ඇන්ටිබයොටික් දාලා බැක්ටීරියා මරන්න. අපි බලාපොරොත්තු නොවෙච්ච විදිහට අඩු කොන්සන්ට්‍රේෂන් එකක් ඇන්ටිබයොටික් එක තිබුණොත් we inadvertently, without knowing that we are doing such a silly thing, we screen, permit, allow the resistant bacterium to grow while all the other bacteria to vanish, perish, destroy. අපි දන්නේම නැතුව අපි antibiotics පොඩි ප්‍රමාණයක් පරිසරයේ තිබීම තුලින් අපි කරන්නේ resistant variety එක promote කරනවා. ඒක තේරෙනවද කියන්න? සිංහලෙන්ම කියලා දුන්නේ. Can you understand the mechanism?
Yes.
Yes.
Yes, sir.
Okay, thank you. That is how evolution happens. Evolution කියන්නේ හරි ලස්සන, very interesting, very truthful principle. There is nothing wrong in the theory, nothing to be improved in the theory. It's always 100% correct. So that is how evolution occurs. The evolution of resistant varieties occurs. So foods, yes, we can apply antibiotics to kill them, but we are not allowed to do so because we know that if it is allowed to occur, allowed to practice, it gives rise to it opens the avenue, opens up the chances for the emergence of resistant variety. The same thing, antibiotics is not allowed. Antibiotics are not allowed to be added to poultry feed, animal feed. If you add in enough quantities to kill bacteria, there is no problem. But if you add the sublethal concentrations of antibiotics, that the problem starts from there. I hope you can understand. If you can't understand, just ask from me. මොකක් හරි පොඩි හරි කොස්සක් තියෙනවා නම් අහගන්න. ඉතින් මේක ඕගොල්ලෝ සල්ලිත් ගෙවලානේ තියෙන්නේ මේ කෝස් එකට. එහෙම නැත්තම් හොඳට තේරුණා කියලා කියන්න මට එතනින් එහාට ගිය හැකියි. ළමයි තේරුණාද?
Hello, sir.
Yes,
ඔව්.
sir.
Yes, sir.
Sir, how
ඔව්
is your
කියන්න.
idea about the freeze drying and freeze concentration when we using the preserved fruit?
Ask it again, dear. I'm very thankful for you to ask for asking the question, but the question I couldn't hear it properly. Please say it again.
Sir, how is your idea about the freeze drying and freeze concentration when we using the food preservation?
Freeze drying and freeze drying is you can call freeze drying for freezing because it does not allow liquid water to remain. It freezes liquid water. So unless the water remains liquid, no organism is able to use it. So it's a sort of drying. So you can call it freeze drying if you want, but there is a separate process called freeze drying that is done to microorganisms, and for food as well you can do it. Freeze drying means you allow the water to be, any organism, any food, anything contains little water, and you allow it to be frozen simply by keeping it in the freezing conditions would make it frozen, right? Then you apply vacuum, then you suck, then you apply using a very strong vacuum pump. You try to suck air out from this. You try to make this chamber frozen. There is a chamber and your frozen food is there, frozen. It has a refrigerator also, refreezer also. So you make it frozen inside the freeze dryer and you apply negative pressure. That means you apply vacuum. The strong pump would try to remove all the air from the chamber. What does that do? That sucks out water without, I don't know the exact chemistry behind this, but that removes water from the frozen condition. I think it makes, now I know what it is. It converts, solid water is ice. Water is liquid water. Water vapor is, you know what it is, Humale. Freeze drying makes ice, that means frozen water, into water vapor without turning it to liquid. Rapid conversion of ice into vapor. Vapor, don't think that vapor is always hot. Water vapor, when we boil water, it becomes vaporized. So that vapor is 100 degrees for sure. But how about water vapor in the atmosphere? Is it hot? No. Actually, when it is liquidized under low temperature, it comes to the earth as rain. So water vapor means not always hot. That means molecules separated. And in the freeze drying machine, frozen water, that means ice, turns into vaporized water and is removed. Steam කිව්වොත් hot වෙන්නේ නේ, vaporized water. You due to that very strong negative pressure, without damaging the texture, without damaging the cells, without damaging the cells, without damaging the food, without damaging the bacterium. It removes water. And there are two things actually. Food, maybe the student who asked the question is concerned about the freeze drying of foods. And I am working in a microbiology department. To my mind, when you say freeze drying, it is freeze drying of bacteria. I will explain, right? In our lab, we make bacterial cultures freeze dried. That means we grow bacteria colonies on agar. We scrape it and collect it and put it in a vial and put in the freezer, put in the freeze dryer. So that makes the bacterial cells and accompanying water, everything frozen. And then we apply the pressure pump, negative pressure pump, vacuum pump. And then the bacterial cells become dry, extremely dry. The water, without damaging the bacterial cell, water goes out as vaporized water. So what we get is a powder, extremely dry powder. There is no water in it at all. So bacteria, are they dead? No, they are not dead. Are they alive? No, they are not showing any metabolism. Can you revive them? Can you give them life back? Yes. If you add nutrients and water, they absorb water and grow into a healthy bacterial population. So that is freeze drying of bacteria. Freeze drying of fruits. We forget about bacteria. Freeze drying of fruits means dehydration of fruits. Dehydration of foods. අඹ කෑල්ලක් දානෝ වරකා දානෝ dehydrate කරනවා. First you freeze them and then remove water as vapor without damaging the cells. So that can keep, that way you get a dry food, and because of it is dry, because the food is dry now, bacteria fail to grow on that. Right? And then you may wonder what would happen to bacteria which have already been there. What can we say? They will also be freeze dried. Unless some water is there, they will not be able to grow. So freeze drying of fruits and vegetables. Any questions of that? අර ළමයා අහපු ප්‍රශ්නේ කියන්නකො තේරුණාද කියලා කොටසක්?
Yeah, sir.
So what is the other part of the question you asked? Freeze drying is clear now. Freeze drying you can apply for foods. You can make the food very dry without changing the texture, without breaking the cells. You can make the food very dry. And frozen food will lose the water. That is freeze drying, and that way is without, as there is no water in it, hardly any water in it, bacteria fail to grow on that. It's dry. What is the other question you asked? I explained how about I explained about preservation of bacterial cultures by freeze drying. That's an additional fact, not related to your question. And then I explained what would happen to bacteria remaining on the food when you freeze dry it. They will also be freeze dried. Then what is the next question you asked?
So idea about the freeze concentration.
That I'm not sure actually what is meant by freeze concentration. Do you have any, can you offer me any explanation on what is that, what that is?
So it's a melt crystallization where the food water is crystallized into the ice crystal, and then the solid ice is separated from the liquid.
Solid ice is separated from the liquid. No, liquid ice is not there anymore. You freeze it first, do you? ඔයාගේ නම කොහොමද ප්‍රොනවුන්ස් කරන්නේ? යුත් කොහොමද?
යුතාරස.
යුතාරා. දුව කියන්න බලන්න දැන් අපි මොකද කරන්නේ? මම ඕක අහලා නැහැ, දන්නේ නැහැ. මයික්‍රෝබයොලොජි මිනිහෙක්නේ මං ආහාර පැත්තට මේ කියන්නකො මොකද්ද කරන්න කියලා ඔය කියන ක්‍රමෙන් ෆ්‍රී මොකද්ද කිව්වේ ඔයා මොන කොන්සන්ට්‍රේෂන්ද? ෆ්‍රීසිං
freeze concentration sir.
ෆ්‍රීස් කොන්සන්ට්‍රේෂන් කියන්නේ මොකක්ද ඇත්තටම අපි ඔක ලෙහා ගන්න බලමු ඔයා දන්න ටික කියන්නකො මට මම චුට්ටක් දාලා එකතු කරලා තේරුම් ගන්න පුළුවන්ද බලන්න අපි දෙන්නට එකතු වෙලා other students also will get the benefit ඒක ආහාර වලට කරන වැඩක්ද?
Yes sir, it's the method of food preservation.
Okay, I'm sorry I didn't know about this. So what do you do to this? What do you do to your food? You freeze it first, I assume. Do you?
Yes sir, I'm not
ඒකට කමක් නැහැ. It's all right. You know it only partially. It's all right. I also know it only partially. Not as much as you know. I guess that it is the same thing. I have a hunch. I feel like it must be the same thing, but you know, it is a parameter. Freezing concentration, is that right? Freezing? No. What is the word you used again? I'm sorry, I keep on forgetting it. මොකක්ද ඔයා කිව්ව?
freeze concentration.
මොන concentration ද?
Freeze concentration sir.
freeze concentration කියලා කියන්නේ අර freeze drying වල පැරාමීටර් එකක් වෙන්න ඇති කියලා මම සැක හිතනවා. අපි චුට්ටක් කතා කරමුකෝ ඒ ගැන. එතකොට අනික් ළමයින්ට තේරුම් ගන්න පුළුවන්. ඔයා දන්න චුට්ට කියන්නකො දු.
Sir, this freeze concentration currently apply freeze limited to fruit juices, coffee, tea extracts, and beer, wine, these beverages. Sir.
Okay. So you remove completely the water content, or you make it completely freeze, or what? Okay, we have a fruit juice. What are we going to do with it?
I think, sir, this is the method of superior production.
So I keep in mind the word freezing concentration, freeze concentration. Freeze concentration and try to find out an answer during the lunch break. How about that? During the lunch break, I would try to find an answer by searching the internet and try to give you an answer. Is that okay? Can you wait until then?
Okay, sir, thank you.
If I wouldn't be able to offer you an answer, then it's my fault. I apologize beforehand, in advance. Calling in advance, I apologize. අර ඒක වෙන්න කලින් දැනටම apologize කරලා ඉන්නවා මේවා ගොඩ යන්න පුළුවන් වෙයි සමහර විට මොකද්ද මේ freeze concentration කිය I feel that it is something like freeze drying වලම කෑල්ලක් වෙන්න ඇති නේද? එහෙම ඔයාට මොකද හිතෙන්නේ? කමන්නේ අපි ආයෙ කතා කරමුකෝ ඒ ගැන.
Okay, sir. Thank you, sir.
Right. දන්නේම නැතුව ගොඩක් වෙලාව ගිහිල්ලා මම මේ බය වුණා එකපාරට 12ට කාලයි කියලා දැක්කම මේ දැන් ওই විදිහට we can, and another thing is canning the foods. Canning the foods, there are so many things to think about. The acid තියෙන food එකක් දාලා can කළ හැකිද? බෑනේ. acid වලට can එක දිරනවනේ. So there are බෝතලයක නම් glass බෝතලයක නම් acid දැම්ම හැකියි. එහෙම ගොඩක් එක එක පැරාමීටර්ස් තියෙනවා. ගොඩක් ඇසිඩ් දාලා ඉවරලා ආහාර කන්නත් බෑනේ අපිට. මයිල්ඩ් ඇසිඩ් එකක් තමයි දාන්න ඕනේ. ඔයාලා කියන්න බලන්න ආහාර ප්‍රිසර්ව් කරගන්න දාන්න හොඳ වීක් ඇසිඩ් එකක්. ඉක්මන් කරන්න. එතකොට මං acetic acid. Very good. Acetic acid. One student gave the answer I expected. Acetic acid is the one of the most widely used weak acids to preserve foods. Acetic acid දාලා තමයි විනාකිරි හදන්නේ. හරි. There is another acid, lactic acid. මං ඉක්මනට කියලා දෙන්න 12යි කාල විතර වෙනකොට ඔයාලට කෑම කන්න 12 මාට 12යි කාලට විතර බ්‍රේක් එකක් දෙන්නම් පැය එක. හරි. Quickly මේක කියලා දෙන්නම්. It is the very interesting mechanism how weak acids can prevent food spoilage. How weak acids prevent food spoilage. Do we use weak acids? Yes. What is the best example? Pickles. Achcharu kiyanne pickles. You boil vegetables. You boil beans, potatoes, onions, beans. And what would happen to them if you don't add vinegar? They would be spoiled within hours actually. If you boil vegetables in the morning, it will be rotten in the afternoon. So you can't keep boiled vegetables more than a day for sure. But if you sprinkle some vinegar onto it, look, you can keep it for 2 weeks, 3 weeks. What is the secret? It's interesting, isn't it? Before going into describing the mechanism of it, I will give you another example. Milk. Milk, is it easily spoiled? Yes. If you draw milk from the cow, if you leave the glass on the table, few hours, maybe 12 hours, 24 hours, definitely it will be completely spoiled. Would you be able to drink it? No chance. Milk is very easily contaminated by bacteria. But you add some lactic acid bacteria to milk. That makes the milk turns milk into curd. You add lactic acid bacteria this afternoon, tomorrow morning you get curd. Curd kiyanne mee kiri kiyanne curd. Mudopu kiri. You can keep it for 1 or 2 days. They make curdled milk, curd in Kantale, in Tissamaharama. Even if they make curd in all the other places in the country, still they are called either from Kantaleo or Polonnaruwa, Kantaleo, Tissamaharama. Those are the 3 places where they make curd. And in other places also they make curd. And even if they make curd in Kalaniya, they call it from Kantaleo, Tissamaharama. That is the trade jibbing, right? Anyway, the point I wanted to make is curd is not easily get spoiled. Curd you can get bus එකෙත් ගෙනල්ලා කන්තලේ ඉඳලත් ගෙනල්ලා ඉවරලා දවසකුත් තියාගෙන කන්න පුළුවන් නේ අපිට. තිස්සමාරාමේ ඉඳලත් ගෙනල්ලා අතින් අතට මාරු වෙලා ලොරි වල ගෙනල්ලා තොග කඩේට දාලා සිල්ලර කඩේට දාලා නිකන් එළියේ විකුණන්න තියලා ෆ්‍රීසර් එකෙත් නේ ෆ්‍රිජ් එකෙත් නෙවෙයිනේ. නිකන් කිරි විකුණන්නේ එළියේනේ සුපර් මාකට් වල දාලා තිබුණාට ෆ්‍රිජ් එකේ. So you can easily understand that the milk is not going to be spoiled easily when you add lactic acid bacteria and allow lactic acid bacteria to grow. Lactic acid bacteria, why do we call them lactic acid bacteria? Because they produce lactic acid. From what? From growth on carbohydrates, proteins, from growth on organic matter, they produce lactic acid. That's why they are called lactic acid bacteria. And lactic acid is the secret behind the food preservation. Acetic acid is the secret behind the preservation of food in pickles. Lactic acid is the secret behind preservation of foods in preservation of milk in curd production. So what is the relationship between, there is a very strong relationship between weak acids and food preservation. Are you willing to listen to it? Do you know the mechanism, by the way? එක ළමෙක් කියන්න බලන්න. යුතාරා ඔයා කියන්නකෝ බලන්න. දන්නවද weak acid හින්දා කොහොමද බැක්ටීරියා මැරෙන්නේ කියලා? බැක්ටීරියා grow නොවි තියෙන්නේ කොහොමද කියලා? ඉක්මනට කියන්න. Vijay Singh, please. Are you going to say something? Oh, කවුරු හරි දන්න ළමෙක් ඉන්නවද කියන්න නැත්තම් නෑ කියන්නකෝ. යුතාරා දන්නවද? මට මේ කයිවාරුවට නොදන්න දෙයක් කියලා දෙන්න හදනකොට කියන්නකෝ දන්නවද නැද්ද කියලා.
No, sir.
No. Okay. Are you willing to listen to this? සතුටුද මේක අහගන්න? ඔව් කියන්නකෝ ඉතින්. නැත්තම් නෑ කියන්න. එපාද කියලා දෙන්න. හරි. මෙහෙමයි වෙන්නේ. Have I mentioned something called aerobic respiration? Have I not told you that that is the fastest way of degrading organic matter? So you can guess that if you can stop oxygen, you can stop aerobic respiration. But how can we stop entry of oxygen into our pickle, entry of oxygen to our milk? We can't do that. But by adding weak acids, we are going to stop what? We are going to stop aerobic respiratory bacteria. We are going to stop the aerobic respiration of aerobic respiratory bacteria. එතකොට අපිට ආහාරයේ ඉක්මනටම නරක වෙන වේගයෙන්ම නරක වෙන බැක්ටීරියා වර්ගයේ growth එක නතර කරගන්න පුළුවන්ද? පුළුවන්. ඔක්සිජන් නතර කළාද නැහැ. වෙන ක්‍රමයකින් aerobic respiration එක නතර කළා. There is another way of stopping aerobic respiration by aerobic respiratory bacteria. By doing so, we can kill aerobic respiratory bacteria, and by doing so, therefore, we can keep the food longer. That is the idea. Now listen how the mechanism is. What the mechanism is. This is how it happens. Organic matter will be oxidized by aerobic respiratory bacteria into carbon dioxide. By doing so, they produce something called NADH, FADH2, NADPH. We call them reduced coenzymes. Don't panic. This is very easy to understand. We oxidize, you had breakfast. You keep on oxidizing this organic matter into carbon dioxide. You don't need carbon dioxide, you exhaled it. What is the importance of doing so? You produced NADH, FADH2, NADPH. We call them reduced coenzymes. What's the point of producing them? You can use them in reduction reactions. More than that, you can use them in something called electron transport chain. You can provide the NADH or FADH2 or NADPH can provide electrons to the electron transport chain. And you know that it is for respiration. Aerobic respiration වලට electron transport chain එකට electron ගෙනියන්නේ කොහෙන්ද ගෙනියන්නේ? organic matter. Actually NAD plus oxidizes the organic matter. Remove the hydrogen from organic matter. The carbon dioxide is released. Carbon dioxide has no hydrogen, only oxygen. So your organic food has a lot of hydrogen, carbon and oxygen, all three. Can I explain it again? You eat organic foods, don't you? What does it contain? Organic matter. What is organic matter? Carbon, hydrogen and oxygen. What do you do to this carbon, hydrogen and oxygen? You oxidize the carbon of it. What is meant by oxidizing carbon? It is the removal of hydrogen from it. hydrogen removal, why do we call it oxidation? Because an electron is also comes with hydrogen. So removing hydrogen from carbon is removing electrons from carbon. Carbon වලින් hydrogen එකක් ගලවලා අහකට ගන්නවා කියන්නේ carbon වලින් ඉලෙක්ට්‍රෝනයක් ගලවලා ගන්නවා කියන එක තමයි. මොකද hydrogen එක ඉලෙක්ට්‍රෝනයත් එක්කම තමයි ගලවලා ගන්නේ. So that hydrogen is now with NADH. NAD plus removed hydrogen from carbon became NADH. So you had your breakfast. Hydrogen of that breakfast is removed from carbon by NAD plus and taken to the electron transport chain. It is in the mitochondrial membrane. Unless you understand this, you wouldn't be able. Once you understand this, you would be able to understand the activity of weak acids. Yes. Sorry about that. So, organic food you eat, you have NAD plus that removes hydrogen from your organic food. We call it oxidation. It is true because you release carbon dioxide, no hydrogen in that. Organic matter, hydrogen was there. Carbon dioxide, no hydrogen is there. No hydrogen is there. That means hydrogen is removed. That is oxidation. And that electrons are taken to the electron transport chain. The ultimate aim is to produce ATP. So this is how it happens, very briefly. Electron transport chain transports electrons along it. Electron transport chain is located in the mitochondrial membrane. I'm describing you the aerobic respiration. Organic food is eaten, NADH is produced. That NADH takes electrons to the electron transport chain. It is located in the mitochondrial membrane. Electrons are transported along the mitochondrial membrane. At the end, electrons are taken by oxygen, producing water. That is the electron transport. What is the benefit of this electron transport? While electrons are transported along the electron transport chain, protons, H+, are taken from the mitochondrial matrix and put it in outside the membrane, outside the mitochondrial membrane. In reality, it is the inner membranal space. So what would happen? Why have you had your breakfast? Part of your breakfast is used to produce ATP. How are we going to produce ATP using your breakfast? Your organic matter is oxidized. Into what? Into carbon dioxide. What's the benefit of making carbon dioxide? No, there is not. It is not the important product. Carbon dioxide is a waste. So what is important in? NAD plus is used for this oxidation. So what is the point of using NAD plus? Yes, it is advantageous. Why? Because it produces NADH. So what is the point of producing NADH? You can take electrons to the electron transport chain. So what is the point of running electron transport chain? There is a benefit. Why? What is the benefit? While electrons are transported along the electron transport chain, protons, H+, are taken across the membrane and left on the other side. So proton translocation happens as a result of electron transport. Along the electron transport chain, proton translocation happens across the membrane. Protons are taken from one side, left on the other side. As a result, protons will be accumulated on one side of the membrane. In the case of mitochondrium, the protons will be accumulated in between the two membranes of the mitochondria, taken from the matrix, put it outside. In the case of a bacterium, aerobic respiratory bacterium, the electron transport chain is located in the membrane, cell membrane, and so protons will be taken from the cytoplasm and left outside in the periplasm, outside the cytoplasmic membrane. So this leads to the accumulation of protons outside the membrane. Accumulation of protons means lot of positive charges and lot of acidity. pH is different. Charges is also different from one side of the membrane to the other side. So outside, more positive charges, less pH. Inside, less positive charges, high pH. Alkaline, acidic outside. Is there a question so far? Is it understood? අනේ උත්තර දෙන්න උත්තර කියන්න. තේරුණාද නැද්ද?
Yeah, sir.
තේරුණා? Understood. Okay. So when we are going to describe the ATP synthesizing mechanism, right? So when protons are accumulated outside the cell, there will be a charge difference and there will be a pH difference across the membrane. So there is a proton gradient, charge difference, pH difference, and we call it proton motive force, and it can be measured in volts. That's the energy difference, can be measured in volts. Even E. coli can develop 0.2 volts. 0.2 volts. That's not a small amount of energy. So that energy is used to synthesize ATP. This is how it happens. There is an enzyme called ATP synthase. And as the name implies, you can understand that the function of the enzyme is synthesizing ATP. That is located in the membrane. So protons are accumulated. ATP synthase enzyme is there. Protons cannot go back through the lipid membrane. Protons cannot go back through the lipid membrane. But protons can go back through the enzyme. So protons will be forced under the proton motive force through the ATP synthase enzyme back into the cytoplasm or back into the mitochondrial matrix. While protons are traveling through the ATP synthase enzyme, the enzyme is not going to remain still. It's going to rotate. It's going to twist. So the enzyme already have binding sites for ADP and phosphate groups. ADP and phosphate are attached to the enzyme. And when enzyme rotates, enzyme changes its 3D shape, the ADP and phosphate groups are forced, otherwise they will be repulsed, forced to come together. They hit against each other, making ATP. That is the ATP synthesizing mechanism. And we add vinegar or lactic acid, it stops this ATP synthesis. And to understand how it stops the ATP synthesis, you have to understand the mechanism of ATP synthesis that I described just now. Can I repeat it very briefly? Organic matter is consumed. Organic matter is oxidized. NADH is produced. NADH is taking electrons to the electron transport chain. The electrons are transported along the electron transport chain to be taken at the end by oxygen. Oxygen facilitates the continuation of the transport. If oxygen were not to be there, there is nobody to accept electrons at the end, so everything will be jammed like a traffic jam. So electrons will not be transported, but luckily oxygen is there to take electrons away from the terminal of the electron transport chain, so electron transport continues. So you keep on transporting electrons along your mitochondrial membrane. Aerobic respiratory bacterium also keeps on transporting electrons along its cytoplasmic membrane. As a result, protons are translocated. As a result, there will be a proton accumulation. As a result, there will be a building up of proton motive force. As a result, protons will be forced through the ATP synthase enzyme. As a result, ATP synthase enzyme is twisted. And as a result, the ADP and phosphate groups bound attached onto the enzyme will hit against each other producing ATP. That is the ATP synthesizing mechanism. Do you have questions? If you say no, I can continue. දැන් ඉතින් මම කොහොමද උගන්වන්නේ කියන්න තේරුණාද නැද්ද දන්නේ නැතෝ කොතනින් කොහොමද මෙතනින් එහාට උගන්වන්නේ?
Yeah, sir, understood.
Thank you. So this is how ATP is produced. And if ATP is not allowed to be produced, then the organism would die. If we cut off oxygen supply, yes. But here we use a different mechanism. We don't cut off oxygen supply. Still we stop the growth of aerobic respiratory organisms. This is what we do. We add lactic acid. We add acetic acid. Whatever, weak acid. And that acid, think about a bacterial cell, aerobic respiratory bacterium. Weak acid it can go through the cell wall. So weak acid would end up in the periplasm. That means just outside the cytoplasmic membrane. What have we added? We added acetic acid. So acetic acid is in an environment now, everywhere there is a H plus proton because electron transport moved protons into the periplasm. So acetic acid ends up in a place where there are a lot of H plus ions. Acetic acid can break into acetate and H plus. Or acetic acid can remain as acetate. It's a weak acid. It's not going to break into H plus very quickly. So it tends to remain as acetic acid, especially if the environment is full of H plus ions. How can you give more H plus to the environment? So acetic acid molecules would remain as not as H plus and acetate ions, but as intact acetic acid molecules, CH3COOH. Can you tell me, is it charged or is there a charge on that molecule or no charge? ඍණ හරි ධන හරි ආරෝපණයක් තියෙනවද ඇසිටික් ඇසිඩ් මොලිකියුල් එක? කියන්න ඉක්මනට උත්තර. No charge. No charge. So the acetic acid molecule is an uncharged molecule. Can it, because it has no charges, can it diffuse through the lipid membrane? Yes, it can. It is not very small, but not too big either. So acetic acid molecule CH3COOH, no charges. It will diffuse through the diffuse කියන්නේ ලොකු concentration එකේ ඉඳලා අඩු concentration එක පැත්තට යන්නේ. දැන් එහෙනම් cytoplasm පටලේ හරහා acetic acid molecules will diffuse through into the cytoplasm from the periplasm end up in the cytoplasm. So your mom has added vinegar and vinegar entered the periplasm, found that there is a concentration gradient and found that it can diffuse through the membrane. It has ended up in the cytoplasm. And in the cytoplasm, what do you think about H plus ion concentration? In the cytoplasm, H plus ions have been removed and put in the periplasm. Aerobic respiration එකේ ප්‍රතිඵලයක් හැටියට electron transport chain එකේ ප්‍රතිඵලයක් හැටියට protons cytoplasm එකෙන් අරගෙන එළියට දාලානේ තියෙන්නේ දැන්. So inside the cytoplasm, the H plus ion concentration is low. What would happen to acetic acid? Acetic acid will be dissociated into acetate and H plus. Now it gives the problem to the bacterium. Outside H plus, inside no H plus earlier. Now acetic acid has entered, broken into acetate and H plus. and increase the H plus ion concentration until the concentration of H plus becomes equal. Is there a proton gradient anymore? Is there a concentration difference, pH difference, charge difference? Nothing. So how can it produce ATP? So the bacterium has lost the proton motive force because your mum has equated the H plus ion concentration outside and inside. So there is no H plus gradient, no chance to produce ATP. The bacterium will die. So aerobic respiratory bacteria cannot grow there. So you may feel like, how about anaerobic respiratory bacteria? Anaerobic respiratory bacteria, to grow there, they have to have nitrate, sulfate, lot of terminal electron acceptors. And there is no guarantee to say that your boiled vegetable is full of nitrate, full of sulfate. So anaerobic respiration also would not take place without a guaranteed way of supplying terminal electron acceptors. So what is left? Only fermentation is left. Can fermentation degrade organic matter completely into carbon dioxide and water? No. Fermentation itself would produce weak acids, alcohols. Are they bad for the food or good for the food? Would they preserve the food more or less? Alcohol වල දාලා තියෙනකොට ආහාරේ නරක වෙයිද නැද්ද? Acid වල දාලා තියෙනකොට weak acid එක නරක වෙයිද නැද්ද? Fermentation would make the food even more palatable. අර කණ්ඩ බැරි vegetables උත් චුට්ටක් ferment වුණාම fermented vegetables maybe taste even better. So fermentation can take place, but it is not enough to degrade the organic matter completely into carbon dioxide and water. So even if little fermentation happens, it's favorable. And aerobic respiratory bacteria would not be able to grow because they lose the proton gradient. They lose the proton motive force. Without that, they can't produce ATP, they can't live. Anaerobic respiratory bacteria, maybe they can grow. They also cannot grow without proton gradient. Anaerobic respiration also would not take place even if sulfate, nitrate, electron acceptors are there. How can they develop the proton motive force? Anaerobic respiration, aerobic respiration, mechanism the same. So anaerobic respiration stopped, aerobic respiration stopped, fermentation may be allowed, but it does not degrade the food in completely. So pickle would last for few weeks. Another. I have explained to you a mechanism which won the Nobel Prize for biochemistry. ඔය proton motive force ඔය කතන්දරේට 1963 Nobel tag හම්බෙච්ච එකක් මම ඔය කෙටියෙන් විස්තර කළේ. දැන් කෑම කන්න පුළුවන්ද කෑම කාලා එන්න එකට? කීයටද? එකයි කාලටද? එකට එන්නකො. Can I give you a lunch break? Ravindu, shall I give them a break? Lunch break?
Yes, sir. Yes, sir.
At
We'll
what
have
time
that.
we are going to start?
We'll start around 1 or 1:15, sir.
1:15, 1 hour. Okay?
Okay,
Okay,
sir.
bye for now. If you have
Okay,
questions,
sir.
please ask once we start again. Hello, good afternoon. Ravindu? Ravindu? Can you hear me? The students, can you hear me? Good afternoon. Uh, so, what I was trying to explain to you that the weak acid, the actually the strong microbicidal action of weak acids, and hope you can understand. If you have any questions, I can answer. If you don't have any questions, have you managed to understand it? The weak acids, maybe it was little difficult for you to understand it right away. I will explain it again. So respiration needs, aerobic or anaerobic respiration needs something called an electron transport. It is a physical structure, electron transport chain, which is located in bacteria in the cell membrane, in mitochondria in the inner mitochondrial membrane. That is electron transport chain. And that electron transport chain has to transport electrons. And while electrons are transported, protons are translocated. And that is very important for the production of ATP. Because the proton translocation makes a proton gradient, proton concentration difference across the membrane. In the bacterium, outside the cytoplasmic membrane, the concentration of protons is high. Inside the cytoplasmic membrane, that is the cytoplasm, the concentration of H plus is low. Due to this difference occurred, due to the difference is established due to the transport of electrons. So, when you add weak acids to the surrounding environment of a bacterium, it loses the proton gradient. This is how it happens. Weak acid molecules go to the periplasm first. And in the periplasm, as there is lot of H plus in the environment, the weak acid, in this case acetic acid or lactic acid, they are not going to be dissociated into acetate and H plus ions, lactate ions and H plus ions. Instead, they remain intact. That means intact acetic acid molecules or intact lactic acid molecules. So, intact lactic acid molecules or acetic acid molecules don't have any charge. They are not charged. So, they are not very small, not very big. Therefore, they can, without a charge, they can diffuse through the lipid membrane into the cytoplasm. And in the cytoplasm, they end up in a different environment. What is the difference? H plus ion concentration is very low because electron transport has translocated H plus ions. The location of the H plus ions was changed from the cytoplasm to the periplasm. Then there are no very many H plus ions left in the cytoplasm. In that environment, the weak acid tends to dissociate it into the ion and H plus, acetate or lactate and H plus. So the importance here is it loses the H plus ions gradient. Inside the cytoplasm, lot of H plus ions are released by the acid dissociation. Disintegration of acid molecules with weak acid molecules with release H plus ions to the environment. That means lot of H plus ions will be released to the cytoplasm. So what happened to the H plus ions difference? The difference is lost. H plus ion difference is no longer be there. Then what happens? Without the H plus ion difference, it will not be able to produce ATP, because it is the H plus ion difference that helps the ATP production. Now, without the H plus ion difference, the organism will not be able to produce ATP anymore. So, without respiration, without respiratory ATP production, respiratory bacteria would not survive. That means aerobic respiratory and anaerobic respiratory bacteria both are not going to survive. So that is how weak acid helps the preservation. Now one student asked from me, what is freeze concentration? I thought it's a parameter, but Ravindu, your course coordinator, he phoned me and he within 5 minutes he found the answer. He says it is a method of freeze concentration, means concentrating fruit juice into fruit pulp by this freezing and thawing, repeated freezing and thawing. So as he explained, what happens is when you freeze fruit juice, you can understand that fruit juice contains sugars, proteins, pectin, fruit cells. Lot of different things are mixed and water is also there. And when you freeze fruit juice, water molecules form bonds with other water molecules, hydrogen bonds. So water molecules are attached to each other, and so when you freeze water molecules, water forms a lattice, free water, and water forms ice. And what about fruit juice? It contains proteins, carbohydrates, pectin, fruit cells, and so and so. And they don't freeze tightly into a structure. Instead, they freeze very slowly. Under water freezing temperature, fruit doesn't freeze that much. And in addition to that, inside fruit cells, the water get frozen, and the freezing increases the volume. You know that if you keep water in a bottle and put it in the freezer, the bottle will be cracked open. The thing is, when you freeze water, the molecular arrangement makes it slightly increase the volume. So inside the fruit cells, when the water is frozen, the fruit cells get burst. They crack open. So fruit becomes more, with cellular structure will be destroyed, and fruit cells become a pulp. Fruit cells become cracked opened. So the texture of the fruit juice is also changed. Become a more like a mashy entity. Fruit cells, due to the freezing of water inside the fruit cell, they get cracked. So because of these reasons, the water in the fruit juice get frozen separately, and the fruit part of the fruit juice get frozen only lightly. So even if you see it as a solid block, water is frozen rapidly, tightly, while fruit is not that frozen. So when you start thawing it again, when you take it out from the freezer and start thawing, what is going to be dissolved easily? The water is going to be dissolved easily. It comes out and you can remove that water. So what have you got? You got the fruit pulp concentrated. If you thaw it again, and if you freeze it again and thaw it again, if you do it repeatedly, the water is easily removed at each step and the fruit becomes more and more concentrated. Actually, it is not a method of food preservation. It is a method of how to make the fruit juice more concentrated, to keep it as a pulp, concentrated fruit juice as a fruit pulp. That reduces the volume, easy to transport, reduces the water content. Maybe it helps a little bit to keep it longer than it becomes like a sugar solution. Maybe it helps the preservation as well to some extent because it is concentrated fruit juice. Under that concentrated sugar concentration, fruit sugars, bacteria would find it difficult to grow because the water content becomes low. In addition to that, it is a method of concentration to make the volume lower and you can transport it easily. And as I can understand it now, it serves as a preservation method as well, because instead of a thin liquid, a diluted fruit juice, it becomes a thick liquid. Earlier we talked about adding honey or salt to keep microbes dry, kill microbes. And here also, when you make a very concentrated fruit pulp, it will, of course, it will keep it little longer. It is not very favorable for microbial growth because the water content is low. The other, per unit volume, the concentration of water is low. It's like a sugar solution. And when a bacterium falls into it, it will be dehydrated because water goes out rather than going inside the cell. So hope you can understand it, and we have to be very thankful to Ravindu, your coordinator, for that, because I was feeling lazy. I was not trying to find out what the reason, but he found it for me, found it for all of us, and we have to be thankful to him for finding that. And it gives you another clue what to do when you come across something you can't understand. So you have a lot of resources, but one thing is, the internet resources are not always very reliable. Only some of them are reliable while some others are not reliable. So what you have to do is to go through several web pages or artificial intelligence that collects the sum of all different web pages, or you can go web pages individually, several web pages, and get the idea. So now we try to talk about foodborne diseases. And as I explained in the morning, food serves as nutrients for bacteria as well. So bacteria can grow in our foods, and when we consume our foods, lot of bacteria would go into our intestinal tract. But first, the food is entering into an acid bath. So the stomach is full of acid, hydrochloric acid, very strong for that matter, very low pH, about 2. So when bacteria enter into the stomach, most of them will be acid hydrolyzed. Like any other food material, we eat foods and foods get hydrolyzed due to acidity in the stomach. So microorganisms face the same fate, but having said that, you have to understand many bacteria would pass that acidity, survive that acidity and go past the stomach and go to the intestine, small intestine and then to the large intestine. So many bacteria are going to die. Few bacteria would enter and colonize in the stomach, and now we are interested in pathogens. Pathogens, only among the bacterial world, only some bacteria are pathogens, and they enter into the intestinal tract and they colonize. As I said in the morning, the already established normal flora would not permit them to establish that easily. But out of competition, if the pathogen wins the competition, they can establish by producing a population in the stomach. 1 or 2 pathogenic bacteria cannot do us any harm. We have mechanisms to fight against them. But if the bacterial population, if the pathogen population is getting increased in size, large population of pathogenic bacteria may cause us trouble. And very rarely, some of them would penetrate the epithelium, enter into epithelial cells, or penetrate through the epithelial cells into blood, or pass through tight junctions and enter into blood. Some of them have enzymes, different toxins to dissolve the cement in between the epithelial cells. So find their entry into blood. But believe me, this happens only very rarely. Bacterial cells entering into blood, entering into epithelial cells happens only very rarely. Then how can they harm us? Sitting there growing there in the intestinal tract, many intestinal pathogens would produce different toxic compounds, enzymes and other toxins. And enzymes, as I said, may dissolve the cement between epithelial cells. And some enzymes would enter into blood. Some toxins would enter into blood. And to make it worse, some toxins like cholera toxin, they can attach onto the epithelial cells by 1/2 of the toxin. They are called AB toxins. There is a part A, there is a part B. And I think from part B, it is attached onto the epithelial cells. So when it is attached onto the toxin, I mean, when the AB toxin is attached onto the intestinal epithelial cells, one part helps the attachment while another part enters into the cell. And they can cause trouble. For cholera, for example, cholera toxin. It is an AB toxin. Part of it is entered into epithelial cells and change the ability of the epithelial cells to exchange ions. So sodium ions are absorbed through the epithelial cells into blood, but this stops the absorbance of sodium ions through the epithelial cells. What stops it? The cholera toxin. And this means a lot of sodium ions are going to be accumulated in the intestinal lumen, actually in the intestinal tract, in the tunnel. And when the sodium ions are accumulated, to compensate it, bicarbonate ions and chloride ions will come into the intestinal lumen from blood. And with that, water will come. Ion concentration of intestinal tract, when it goes up, the water concentration obviously goes down. Therefore, water moves from high concentration to low concentration, in this case, from blood to the intestinal lumen. And chloride ions, bicarbonate ions also, to make the salt balance, they also come to the intestinal lumen. That means the intestine has lot of ions. That means less water. So water comes along the concentration gradient from blood to the intestinal tract. So that means your intestinal lumen becomes very watery. And so nature has made it in such a way that watery stools have to be removed from the intestinal tract. So you get diarrhea. So the bad thing is, when you have diarrhea, the good thing is bacteria will be flushed away. Bad thing is water will also, your blood will also lose lot of water, and that makes you dehydrated, and that makes the heart fails to pump, and that means dehydration, instant death. The blood volume, if you loses the blood volume, that brings in death more than earlier than you imagine. You think water වතුර පෙවෙන්නේ නැහැ, වතුර ටිකක් බොන්න ඕන තමයි කිය ඉන්නෝ. ජීවනී බොන්න ඕනද? ඔව් කිය බන කිය ඉඳද්දි දන්නේම නැතුව මැරෙන්න පුළුවන්. So if you have diarrhea, just remember to drink water. And if you, I mean, it's a psychological thing. I can't drink water. If you don't want to drink, how can you drink? The pharynx will stop swallowing water. If you don't want to drink it, psychologically, your pharynx will listen to your mind and stop you drinking. You can't swallow it unless you are ready to swallow it. You can't swallow anything unless you are ready to swallow anything, things. So if you don't want to swallow water, you can't swallow water. So you say, you keep on complaining, I can't drink water. That is your mind, not your throat. So if your mind is ready to take water, you can drink water. Why not? I drink 1 liter at a time. A lot of water at a time. If I keep on drinking, I drink water, right? At least a liter in one go. If the cup is full, if the mug is full, if the big jug is full, I drink everything. If I drink from a water bottle, I drink a whole bottle. So if you want, you can drink water. And if you are reluctant to drink water, your pharynx is not ready to drink water, so you can't drink water. So you have the option of dying. If you want to save your life, you drink water. If you can't drink it in one go, you start sipping. 1 liter in one go, or 1 liter in 100 sips. Same thing. So you keep on sipping water. I චුට්ට බොන්න. ඒක බීලා මැරෙයි කියලා හිතෙනවනම්. එතකොට ජීවිතේ බේරගත්තා. ඒක නැත්තම් බේත් නෑ මේකට. බෙහෙත් කියලා જાතියක් නෑ. When it happens, you get a diarrhea. There are medicines to stop diarrhea. What does that mean? That means your pathogen is going to remain in your stomach. Are you happy about that? No, you don't want that to happen. You want the natural mechanism, you want to allow the natural mechanism to flush out the bacteria. So that's the only way. And keep on drinking. Don't forget to do that. You keep on drinking water, not alcohol. You keep on drinking water, and that keep you alive no matter how many times you go to the toilet. Unless your intestinal tract is damaged and you are losing blood, bloody diarrhea. Watery diarrhea cannot kill you as long as you keep on drinking water. But bloody diarrhea you may lose blood and it will bring you death. In that case, you have to seek medical help. But watery diarrhea is not a matter to be worried that much, as long as you keep on sipping water. If you don't want to do that, get admitted yourself to a hospital and get IV fluids. IV fluids that will keep you alive. The best thing for you to do is IV fluids. Nobody knows what is the exact amount you need. Maybe too much fluids also is not going to do any good to you. So IV fluids is an uncontrolled injection of fluids into blood. Oral fluids, the body knows what to absorb and what is the limit of absorbance. So the body would stop it and would not never absorb too much water into your body. So oral hydration is the best method to avoid dehydrogenation. So different bacteria are there, different types of toxins they produce, and sometimes the bacterial parts also. Now I am going to talk about something else, right? Some parts of bacteria can also be poisonous to you. For example, gram-negative cell walls of bacteria. Some parts of gram-negative cell walls can do you harm. If they enter into your bloodstream, it can be even fatal. So bacteria, even if you kill bacteria, their parts can give you trouble, but these lipopolysaccharides, we call them, lipopolysaccharides in the bacterial cell wall, gram-negative cell wall, can cause you trouble, but not mainly in the intestine. If you inject them into your blood, that brings in death. So if you kill bacteria, but if the parts of bacteria, cell wall parts remain and should not be remained in the injection water, for example, that can kill you. So injection water has to be, obviously you know that it has to be without bacteria. Now you know that it has to be without bacterial cell wall components as well. So dead bacteria, but their cell wall components should not be allowed to remain in whatever injected into your blood. So bacterial growth in the intestine, Vibrio cholera, Salmonella, Shigella, so many different bacteria are there, can cause intestinal infections, but to different Staphylococcus aureus, they can cause trouble, but most of the time these trouble are not fatal, cannot bring you death unless you keep on, as long as you keep on rehydrating your body, it's difficult to kill you, but still sometimes it may become fatal. Sometimes the toxins can enter into your blood, septicemia. That gives you a serious problem. Give a systemic inflammation, your body tend to lose lot of fluids, and so many different problems can be brought, but bacteria are there, as I said. We have been there. So humans survived without knowing any microbiology, without having any antibiotics. Normally, stomach problems, antibiotics are not given, no needed to give. Diarrhea is the God-given method. Diarrhea is a blessing. And avoiding too much diarrhea and avoiding dehydrogenation is the best thing to do. And now I am going to talk about something else. If you ingest bacteria, pathogenic bacteria, very clearly you can call it a food infection, foodborne infection or food infection. Food cannot infect you, but by saying food infection, what you mean is foodborne bacteria, pathogenic bacteria, foodborne infection. So we call it food infection. Another thing is some bacteria. What happens if they grow in your food and excrete some toxins while growing in the food? And you don't need to engulf a huge population of bacteria. Instead, a drop of this toxin would be enough to kill you. You are not going to wait until the bacterial population will grow in your stomach and excrete toxins, excrete enzymes, and giving you harm. Here, in a different situation, the bacteria already grown in your food and the toxin they excreted is enough to kill you. You don't need to wait until the bacteria grow in your stomach. So you can clearly understand that antibiotics are definitely going to fail in this situation, because you don't expect the microbial growth in your stomach. Even if you stop the microbial growth by taking antibiotics in the intestinal tract, you stop the growth of microorganisms in the intestinal tract by taking antibiotics. But the bacterium has already grown in your food, secreted some toxins already, and you are ingesting food with the bacterial toxin. How can you call it? Can you call it a food infection? Do you expect bacteria to grow there and go there and grow there, establish there as a population? No need. The toxin is already in your food. So we can't call it bacterial infection. Food infection, foodborne infection, you can't call it. So what is the name you can call it? We can call it, can we not call it intoxication? We can call it food intoxication. You are consuming toxins, food intoxication. I will give you a classic example. Clostridium botulinum. That is an anaerobic respiratory bacterium. Sorry, not anaerobic respiratory. I am really sorry. It's a fermentative bacterium, but it is an anaerobe. Anaerobe means cannot live in the presence of oxygen. It is killed in the presence of oxygen due to the mechanism I mentioned in the morning, due to the production of reactive oxygen species and absence of anything to react against reactive oxygen species. No antioxidant mechanisms. So, Clostridium botulinum can grow as an anaerobe. It can grow in a tin can. If it has entered, if your sterilization didn't work, you can the food, and here the bacterium find an ideal environment. What is ideal for its growth? Because it is anaerobic. The can is completely anaerobic. Even if some oxygen is there, it will be finished by aerobic respiratory. If some aerobic respiratory organisms left, they would finish oxygen. So in the can, you can't expect any oxygen to be there. So that's the ideal situation for Clostridium botulinum spores to grow. If your sterilization process was not very successful, if a spore accidentally is left in the can without being killed, so the spore will germinate under anaerobic conditions, and they would, as a matter of fact, Clostridium botulinum is a fermentative organism, so it keeps on fermenting the organic matter. So during fermentation, it may release some gases as well, carbon dioxide, hydrogen. So these gases are accumulated inside the can. So if the can is bloated, you can suspect that there is some bacterial growth releasing some gas. And if that bacterium were to be Clostridium botulinum, it must have secreted enough toxin to kill even an elephant. It's a very strong neurotoxin, which is called botulin. So botulin can be there, produced by Clostridium botulinum in the absence of oxygen in this can. So what would happen if you eat it? Even tasting it is enough to kill you by paralyzing your nervous system. your nervous system needs from the brain. If you want to constrict a muscle, you have to send a signal through the motor neuron. And if motor neuron sends a signal to the end of the neuron, the signal has to go to the muscle. So the motor neuron ends up close to the muscle fiber. And the motor neuron secretes acetylcholine, a biochemical, to trigger the constriction of the muscle fiber. What happens if you have botulinum in your system? Botulinum would go and jam the nerve ending. That means cannot secrete acetylcholine anymore. So as a result, the muscle would not be able to constrict it. So if I want to constrict it, I can do it immediately. Constriction of skeletal muscles. But if you cannot constrict skeletal muscles, you cannot move the muscles. You cannot do anything using muscles. So you can't lift your hand. You will be paralyzed. And what you get is muscle paralysis. Muscles cannot be constricted. So what would happen if your rib cage, the muscles in the ribs cannot be constricted? You cannot breathe. If you cannot breathe, what does that mean? You are going to die. So Clostridium botulinum, the bacterium, which is an anaerobe, that means can live only in anaerobic conditions. So in the tin can, it finds a better environment. If the sterilization technique was not successful, if one or two spores to be remained there under anaerobic conditions, they can go into a population and they would produce neurotoxins in the can. You don't need to ingest a bacterial population. Just the taste of the bloated can is enough to kill you. That means botulinum, the very strong neurotoxin, will enter into your body. Other than that type of bacteria, there are hardly any bacteria which can kill you, but it's a nuisance. You get a stomach upset, you get few rounds of toilet, watery diarrhea, and soon you will recover as long as you take water. Otherwise, that can bring you death. That's about food infections and food intoxications. So no need to grow bacteria. Even in the refrigerator, Staphylococcus aureus, it is occupied. It is a normal bacterium living on your skin and in the nasal cavity. And the person is not going to, he is only a carrier. It's not going to get any problem as long as it lives in the nasal cavity. But when you scratch your nose and make a salad, Staphylococcus aureus can enter into your salad and you keep it in the fridge. But 4 degrees of centigrade cannot stop the growth of Staph aureus. It can grow there and it can secrete some toxins also. And when you eat the salad, you eat toxins, you eat the bacterium, and it can establish in your stomach and it can give you fever and diarrhea, but maybe not going to kill you. So there are bacteria like that. Each and every bacterium has its own severity, virulence. So different virulent factors are there. Even the flagellum can be toxic. The enzymes, toxic. Enzymes degrade tissues. Toxins act in many different ways. Some of them are pyrogenic. Some toxins can control the thermal regulation, upset the thermal regulation in the body. So you get fever, temperature. So there are a lot of mechanisms depending on each bacterium, and when you need to know about each and individual bacterium, you please find some resources in the internet. Staphylococcus aureus, how can it bring, what type of stomach problems it can bring about. So it will give a lot of information, and I don't need to waste time on describing these things. So food infections have to be prevented. Have to keep in mind that even if you don't allow infections, intoxication can happen, and that's bad for you. So water also. Waterborne diseases. Waterborne diseases is a problem. It's not separated from food microbiology, because waterborne pathogens can bring foodborne infections in the stomach. You don't need to eat the bacterium with the food. Some people, when a person suffers from a stomach infection, listen carefully. When a person suffers from a stomach infection, food infection, there will be pathogenic bacteria in his fecal matter, his or her fecal matter. I told you, diarrhea comes as a means of getting rid of the bacterium, flushing it out. So when you have diarrhea or when you defecate, lot of intestinal organisms will go out with fecal matter. If you are suffering from a intestinal infection, your stools are full of these pathogenic bacteria. What happens when this pathogenic bacteria enters into your food? Of course, a food infection. What would happen if the pathogenic bacterium enters into a water body, a river? Are they going to remain in the water forever? Not really. Pathogenic bacteria need lot of nutrients. Their natural habitat is the intestine. So they are not going to survive too long in water, maybe few days. Is it good to drink that water if they are there? If they are dead, once they are dead, it's okay. The water is okay to drink. If they are not dead, in the first few days of entering into water, they will not die. Drinking that water will definitely give you a food infection, stomach infection, intestinal infection. So how to avoid them? You have to see whether this water is contaminated by fecal matter or not. If the water has the water got pathogenic bacteria or not? Given the variety of bacteria which can cause diseases, and fungi and viruses, given the wide range of the diversity of organisms and viruses that can cause you disease, it's very difficult for you to find out, to do tests for each and every pathogen, to find whether salmonella is there, to find whether a virus is there, to find whether Shigella is there, to find whether Clostridium is there, find whether Listeria is there. So it's not going to help, it's not going to work, because you have to drink water, especially the water distribution facilities, municipal water distribution centers have to release this water every day. So many liters of water are consumed. So do you have time, money, resources to test for every single pathogen on earth in this water? The answer is no. So what are we going to do? Just to release the water, hope for the best. Make a නැතුව ඇති bacteria ඔය එකෙක් දෙන්නේ මැරුණාම ආරංචි වෙයිනේ. එහෙම you can't do that either. So what are you going to do? Luckily, there is a bacterium called E. coli, Escherichia coli in our intestinal tract, in everybody's stomach. Whether suffering from an intestinal infection or not, every one of us, every human being, Donald Trump, Netanyahu, everybody carries this E. coli bacteria, Escherichia coli, in the intestinal tract. So every time you defecate, release the fecal matter, billions of E. coli are going to escape from your going to be released from your body, and they can enter into foods, they can enter into water bodies. If they enter into foods, they will grow there. Sorry about that. So, where were we? So, pathogenic bacteria is a nuisance, but you know what to do. And each and every different. So I was talking about water. So when a pathogenic when pathogens enter into a water body, I told you they can live for few days depending on the nutrient content of water. Some water has some organic matter, some water has very, very little amount of organic matter. So can't say how many days, but within few days, the pathogens would die. Similarly, within few days, E. coli also would die. Your fecal matter, my fecal matter, anybody's fecal matter contain E. coli. Only infected people's fecal matter got pathogens. So how can we find out whether there are pathogens, whether there is a risk of having pathogens in this water? All you have to do is to check for E. coli. E. coli goes out with fecal matter from anybody's intestinal tract, can grow a few days in water, can survive a few days in water and then die. Pathogens enter into water bodies, can enter into water bodies with fecal matter of infected people with E. coli. E. coli is always there. Pathogens are there only when the person is infected. So can enter with E. coli and not going to remain forever. E. coli is also going to die. Pathogens are also going to die within few days. So if E. coli is not there, what can you say? Pathogens are not there. If E. coli is there, what can you say? Pathogens are there? No. Pathogens could be there. There is a risk. So we call the E. coli organism the indicator organism of fecal pollution of water and foods. If E. coli is there in water, that means recent fecal pollution. If there is E. coli in your food, that shows the risk of that shows that it is contaminated with fecal matter, and that shows the risk of E. coli නිකං ආහාරවල හැදෙන්නේ නැහැ, නිකං වතුරෙත් ඉන්නේ නැහැනේ. So it is a sure indication of fecal contamination. But it is not a sure indication of having pathogens. Still, it is an indicator of the risk of having pathogens. දැන් පත්තරේ දැකලා තියෙනවද අසූචි මිශ්‍ර ආහාර විකුණපු වෙලෙන්දාට දඩ කියලා? Do you think that fecal matter is found in food? No, no, never. They say so because it indicates fecal pollution. The presence of E. coli is what they have found. E. coli is in this food. E. coli is in this water. Does it say that it indicates fecal pollution? Yes. Does it say that it indicates recent fecal pollution? Exactly. That is what it indicates. Does it indicate the presence of pathogens? No. Does it indicate the risk of the presence of pathogens? Yes. සමහර විට pathogens ලත් ඉන්න පුළුවන්. E. coli නැත්තම් ඉන්නම බෑ. E. coli ඉන්නවා නම් pathogens ලත් ඉන්න පුළුවන්. ඒ කියන්නේ වතුර බොන්න හොඳද? E. coli තියෙනවා නම්. කෑම කන්න හොඳද? E. coli තියෙනවා නම් කෑමවල. නෑ. ඇයි E. coli වහින්දද? නෑ. E. coli අපි හැම කෙනාගේම බඩේ ඉන්නවා. E. coli innakota, it indicates a risk of the presence of pathogenic organisms. Hope you can understand it. And I will very briefly tell you how to find whether E. coli is there or not. Testing for E. coli, testing for different pathogens is practically impossible, I told you. Testing for E. coli is relatively easy, but in the first round, we are not going to look for E. coli. Instead, we look for the presence of coliforms. Who are coliforms? There is a group of bacteria. there is a group of bacteria in this world, they have a common character. They can ferment lactose. They produce gases, hydrogen, carbon dioxide during this fermentation of lactose. And they can do it within 37 to 48 hours at 37 degrees. This group of bacteria sharing these common characteristics I explained to you, they are called coliform group. Coliform group of bacteria. Who are they? They can ferment lactose, producing acid and gases within 37 to 48 hours, within 24 to 48 hours at 37 degrees. That is the characteristic of coliform group. And E. coli is a bacterium which belongs to this coliform group. So, E. coli is a coliform. Coliform is a group of bacteria. And actually, it's easier to, the testing for coliforms, the whole group, is easier. All you have to do is to find out whether they can produce gas and acids upon growth upon lactose within 24 to 48 hours. That's easy to find. E. coli, testing for E. coli testing for E. coli takes time. So what's the point of testing for coliforms? If coliforms are there, some coliforms are not even intestinal. They live in soil, environmental organisms. So does it indicate anything? The coliforms? No, it doesn't indicate anything. Then why do you do coliform test instead of doing E. coli? E. coli is the indicator organism. You have to find out whether E. coli is there or not. Instead of looking for E. coli, you look for coliforms. Why? Because coliforms are also only very rarely present in water. So if coliforms are not there, you can release this water for consumption. You are doing the coliform test hoping that there will be no coliforms. Actually, as a matter of fact, most of the time coliforms are absent. So do you need to do tests for E. coli? No. The simple coliform test, you can do it. You may be working in different organizations. Your place of work or the place of university, place of study. If I go there to your company and ask from different people whether Samadhi is there or not, it takes time. Instead, I can phone the company and say, are you working today? If they are not working, I know for sure that Samadhi is not there. So I can save energy, save money, save time by looking for an individual. Instead of looking for an individual, you just find out whether the group is there or not. Instead of looking for E. coli, you can easily find out doing the test for coliform group. You can easily find out whether there are coliforms or not. This is not going to work, not going to be a bright idea if coliform test becomes all the time positive. කොච්චරවත් coliforms ලා ඉන්නවා. එහෙමනම් coliform test කරන එකේ වැඩක් නෑ. ඊයේ වැඩක් නැති. Then you have to do for E. coli test. So what's the point of wasting money and resources. So, but the good thing is, most of the time in the practical world, coliform test gives negative results. So you can call it a day. You can say, no, we don't need to look for E. coli. Coliforms are also not there. You study in a faculty of science in the university, the Department of Microbiology, Faculty of Science, University of California. So the largest organization is University of California. Instead of finding out whether microbiology students are there in the university today or not, you can either go to the university and each and every student you can ask, are you doing microbiology? Or you can phone the university security services and ask whether the students are there in the university or not. If students are not there, there is no chance for microbiology students to be there. If coliforms are not there, there is no chance for E. coli to be there. So you can release this water. So cut lot of work short, you test for coliforms first. And then if they are there, the matter becomes serious. Then you have to study for, then you have to look for E. coli. Still you don't start testing for E. coli first. You know that E. coli can withstand 44 degrees of centigrade, 44 degrees. It lives in an intestine 37 degrees and 44 degrees is not going to do it any harm. So you test, you see whether this organism, whatever coliform present, instead of trying to see whether it is E. coli or somebody else, you test whether it can grow at 44 degrees or not. If it can grow at 44 degrees, it looks like E. coli. If it cannot grow at 44 degrees, you know for sure that it can't be E. coli. So still, the second test is called fecal coliform. The fecal coliform is also a group, subgroup of coliforms. So fecal coliform, if they cannot grow there, if they are not there, you know for sure that E. coli is not there. The problem starts if fecal coliform test, that means at 44 degrees. If it gives positive results, fecal coliform test, positive results are given by E. coli. Not only E. coli, Enterobacter also gives positive results. What is Enterobacter? Is it a fecal organism? No, it is not a fecal organism. It's a soil organism. But we call it a fecal coliform only because it can show the characteristics of fecal coliforms, characteristics of E. coli. So if from fecal coliform, we don't know whether it is E. coli or Enterobacter or some other bacteria. We don't know. But if the fecal coliform test, 44 degrees test is positive, then we need to do further tests to confirm whether it is E. coli or not. And there are some other methods. If the fecal coliform test is positive, you have to know for sure whether it is E. coli or Enterobacter. And there are several tests: indole test, methyl red test, Voges-Prosker test, citrate test, growth of bacteria on EMBA, colony appearance. So many tests are there to distinguish between, to know for sure whether it is E. coli or Enterobacter. So that is water testing. And not only that, there is another important thing. The bacterium E. coli, if one E. coli in the entire Kalni River, does it indicate recent fecal pollution? No. E. coli has to be there in 100 milliliters of water. If the river water contains, if the water contains more than one or more E. coli, that is the internationally accepted standard. If there is a single E. coli or if there are more E. coli in 100 liters of water, then that is what indicates recent fecal pollution. නැතුව වතුර බැරල් එකක් ගඟෙන් ගත්තාම එකෙක් දෙන්නේ කීකෝලා හිටියයි කියලා ප්‍රශ්නයක් නැහැ. It does not indicate fecal pollution. It is not a dangerous level that indicates dangerous fecal pollution. ඔය කොහෙන් හරි කවදා හරි කුරුල්ලෙක්ගේ මේ වලින් තිබිලා හරි E. coli ටිකක් එන්න ඇති කියලා හිතාගෙන නිකා හිටියකි. But if there are E. coli 1 or more in 100 milliliters of water, that is accepted internationally as the indication of recent fecal pollution. Now you can understand that not only the presence or absence, the number is also involved. Number is also matters. Number of E. coli. How finding them itself is difficult. Counting the numbers, especially when the number is so low, 100 milliliters just a single bacterium. අපි කොහොමද බලාගන්නේ මිලිලීටර් 100ක මේ වතුරේ එක E. coli කෙනෙක්වත් නැහැ කියලා. It sounds difficult. No other method is able to find out whether a single bacterium or 2 bacteria are there in or not. So there has to be a very sensitive test to find out that තේරෙනවද ඔයගොල්ලන්ට මේක? දැන් මම දිගටම කියවන යන පැයක් විතර. දැන් කියන්න බලන්න තේරෙනවද කියලා. එක්කෙනෙකුටවත් තේරෙන්නේ නැද්ද? උදුම ළමයිනේ කාලනාස්ති කරවනේ. It's your money, your time. Why can't you have the courage to say yes or no? Disgusting. Foolish. Dulen. Okay. So finding out whether E. coli is there is E. coli or not is not the final thing. You have to count the bacteria and look at this water sample is there, river water. Can you see E. coli in there? No. Is there a way to find it out? There is only one way to find it out, and the count then. Counting is also important. Counting. So I will invite you to think. Without thinking, you can't understand this. And without answering also, you can't understand it. I expect you to at least 1 or 2 students to answer me verbally. Otherwise, I will stop this in the middle. Now, I am not going to ask a very serious question. You want to count the number of, you want to see whether E. coli is there in this water or not. In the same time, you want to count the number. Sounds a difficult job, but I am going to explain a very easy method. So how are we going to find out? You are given a beaker of water, asking you to find out whether there are E. coli cells, 1 or more, in this water or not. You don't know how to do this, but now there is a way. I will tell you. E. coli can grow, even one cell of E. coli can grow in a test tube to produce a big colony growing on lactose. Produce a big colony to produce a visible result. Acid production and gas production. Both can be visible. Acid production can be detected by using a pH indicator. Gas production can be detected by putting a small tube upside down in the medium. So you take 10 milliliters of river water and add to this medium, lactose broth. And in the same time, you take 1 milliliter of the medium, you take 10 milliliter of the medium and add 1 milliliter into it. And you take another tube and add 0.1 milliliters to it. You have 3 tubes of the medium. You add 1 milliliter, 0.1 milliliter and 10 milliliters into these 3 tubes. And then you put this in the incubator, knowing that if at least if 1 E. coli cell is there in the river water, transferred into the medium, it will grow into producing a visible result. So you come in the following morning. All 3 tubes showed no growth. What would be your conclusion? What are you going to say? එකකට මිලිලීටර් 10ක් දැම්ම ගඟේ වතුර. අනිත් ටියුබ් එකට එකකුයි අනිත් ටියුබ් එකට දශමයකුයි දැම්ම එකකවත් growth එකක් නැහැ. මිලිලීටර් 10ක්ම දාලත් ඒක එක සෛලයක්වත් නැහැ කියලා කියන්න පුළුවන්ද? Even if I have added 10 milliliters, it did not show any growth. That means even 10 milliliters of that water did not carry a single bacterium. තේරෙනවා නම් නිකයින්ද තේරෙන්නේ නැත්නම් අහන්න. හරි නැත්නම් කරන්න දෙයක් නැහැ. මට පොත්වලත් නැහැ මේක එහෙම ලේසියට පැහැදිලි කරලා. If you come on the following day, if there is no growth in any of the tubes, you can say even 10 milliliters of the river water does not contain a single, at least a single E. coli. If all 3 tubes show positive results, what can you say? Tube 3ම දැන් positive results පෙන්නනවා. එතකොට කිව්ව හැකි even 0.1 milliliter, a very small amount of this river water contains at least 1 bacteria. ඒකත් better than nothing. It's something about numbers. මිලිලීටර් 10කවත් එකෙක්වත් නැහැ කියන එකකුත් නම්බර් එකක් නේ. මිලිලීටර් දශම එකකත් එකෙක් හරි ඉන්නවා එකෙකුට වැඩියෙන් ඉන්නවයි කියන එකත් නම්බර් එකක්. Better than nothing. We are getting closer to the actual number. And if the scenario is something like this, 10 milliliters, you added 10 milliliters of water, no growth. 1 milliliter, again growth. 0.1 milliliter, no growth. What are you going to say by looking at the 0.1 milliliter tube? I mean, the tube you added 0.1 milliliters of the river water. There is no growth. What can you say? කරුමයක් නේ මේක උත්තර නොදී ඉන්නේ. උතුම කරුමයක්. උදේ කතා කරපු ළමයි කවුරු හරි කතා කරන්න. මේ 0.1 milliliters of river water didn't show any growth. What can you say? Pavindu, what should I do?
Yes, sir. Yes, sir.
What can I do now? These idiots chose to stay silent. මිලිලීටර් දශම එකක් දාපු එකේ මොකක්වත් grow වෙලා නැත්තම් මොකක්ද හරකුනේ කියන්න පුළුවන් ඒකට. Hello. උදුම කරුමයක් නේ.
If you can drop a message, even just do it.
කටවල් කැඩිලාද? උදුම කරුමයක් කාලකන්ණි සල්ලිත් ගෙවලා ඉන්න විරිත්තගෙන. දශම එකක් දාපුහම ගඟේ වතුර ඒකේ growth එකක් නැත්තම් මොකද්ද කියන්න පුළුවන්? දන්නේ නැද්ද? ඔළුවේ නැද්ද? ඒ දශම එකේ යකෝ එක බැක්ටීරියාවක්වත් නැහැ කියලා කියන්න බැරිද? පුදුම හරක්නේ කතා කරන්නේ නැතුවම මට පුදුම තරහා යනවා. පුදුම කාලකන්ණි සල්ලි ගෙවලානේ මේ එකට ආවේ. Ravindu, you tell me. You have added, Ravindu,
Yes, sir.
I have added 0.1 milliliters of river water, and it did not show any visible growth. What can you say? Can you not say the 0.1 milliliter did not carry a single cell? මුන්ට තේරෙන්නේ නැහැ මේක
Yes.
අහගන්න නැතුව හිතන්න නැතුව තේරෙන්නේ නැහැ නේ ඉබ්බෝ වගේ මෙතෙන්ටලා ඉඳගෙන හිටියයි කියලා. So
Yes.
can you what can you say? The absence of growth in the tube into which you added 0.1 milliliters definitely show that 0.1 milliliter does not carry a single bacterium. ඒක හරිද?
Yes.
By looking at the next tube, you added 1 milliliter. It showed some growth. What can you say by looking at that tube? 1 milliliter.
The contents.
1 milliliter contained 1 milliliter of this river water contained at least one bacterial cell. Can you not say that?
Yes.
If you look at both tubes, the presence of growth, the absence of growth. 0.1 milliliter, no growth. 1 milliliter growth. By looking at both tubes, can you not say that 1 milliliter of this river water carry at least one cell, but that
One does not contain.
that number cannot exceed 9.
Yes.
If it were to be 10 or above, 0.1 or 10 times less volume may carry at least 1 milliliter එකක 9ට වැඩි ඉන්නවා නම් මිලිලීටර් 0.1ක එකක්වත් ඉන්න එපායි.
Oh.
එතකොට මිලිලීටර් 0.1ක් දාපු ගෝ එකේ ග්‍රෝ වෙලා නැහැ. මිලිලීටර් එකක් දාපු එකේ ග්‍රෝ වෙලා තියෙනවා. දැන් කියන්න පුළුවන් අපි පියවරෙන් පියවර ගියොත් 0.1 නම් එක බැක්ටීරියාවක් ඉඳලා නැහැ මේකේ growth එකක් නැහැ. මිලිලීටර් 1 දාපු එකේ grow වෙලා. ඒක දිහා බලලා කියන්න පුළුවන් මේ මිලිලීටර් එකක එකෙක් හරි ඉඳලා තියෙනවනේ කියලා. දෙකම දිහා බලලා කියන්න පුළුවන්. මිලිලීටර් එකක ඉන්න ගාන 9ට වැඩි වෙන්න විදිහක් නැහැ. ඒකේ 10ක් හිටියා නම් මිලිලීටර් 0.1කට එකෙක් හරි එනවනේ. 10න් බෙදපුවම.
Yes. Ratio එක නේ.
That is what we call the most probable number. Most probable number, MPN, of the bacteria. We can increase the accuracy of this. Why not? Taking instead of 10 milliliters into one tube, we add 10 milliliters separately into 5 tubes. 1 milliliter into 5 tubes. 0.1 milliliter into 5 tubes. So altogether we have 15 tubes. And according to the result, there are statistical tables, MPN tables. You can get a number. That is, I have explained to you the principle of this MPN method, which is hardly explained in books. මොකෙක්වත් ඕක explain කරලා නැහැ හරියට පොතක. හරි. ඔය මිලිලීටර් 10ක් දාපුවම growth එකක් තියෙනවා, එකක් දාපුවම growth එකක් තියෙනවා, 0.1ක් දාපුවම growth එකක් නැත්තම් අපිට මිලිලීටර් එකක් දාපු එකේ එකත් 9ත් අතර බැක්ටීරියා ගානක් ඉඳලා තියෙනවයි කියන්න මේ ගඟේ මිලිලීටර් එක බැක්ටීරියා එකත් 9ත් අතර ඉඳලා තියෙනවයි කියන්න පුළුවන් නේ. ඉතින් මේ ළමයින්ගේ කරුමෙට මේ ළමයි අහන්නේ ඕක විස්තරේ. දැන් ඉබ්බෝ වගේ ඉන්නවා සල්ලිත් ගෙවලා මේ කෝස් එකට සද්ද නැතුව. මරන් කනවද? වැරදි උත්තරයක් දුන්නත් චුටක් හිතන්න පොළඹවන්න හැදුවේ. ඒක තමයි ඉගෙන ගන්න තියෙන හොඳම ක්‍රමේ. හිතන්න නැත්නම් ඉතින් මම මැරෙන්නද? ගෙදර ඇවිල්ලා කියලා දෙන්නද? මේ හොඳට උදේ කතා කරපුවේ වුණොත් දැන් බය වෙලා ඉබ්බෝ වගේ ගෙම්බෝ නම් කෑ ගහනවා. ඉබ්බෝ වගේ ඉන්නවා. ඉබ්බෝ. Right. That is, if you don't ask questions, I don't have any appetite to tell you. In the morning, somebody asked an interesting question, not only one, few questions, and that helped me to answer, and that supported other students as well. I don't know this habit. උදුම වැඩි හිටියෝ නේද කසාද බැඳලා ළමයි එහෙම කවදාවත් ළමයිවත් හදන්න එපා කතා කරගන්න තරම් කොන්දක් පන්න නැත්තම් එච්චර දුර්වල නම් මානසිකත්වය මේ මැරෙන්න ගඟට පයින්න. I'm sorry to say this, but since in the morning I warned you so many times. In the beginning, when I started this explaining this, I asked you to say at least yes or no, but you chose to remain silent. I really don't like this. Anyway, Ravindu,
Yes, sir.
I have to explain these idiots, these dumb people, what HACCP is, HACCP. Are you willing to
Yes, sir.
support? Huh? Are you willing to support me? Are you willing to help me to teach these people?
Yes, sir. Yes, yes.
So food processing plants are there. Food industry is a big industry. You have to make sure that the food does not contain microorganisms. You have to make sure that food does not contain gravel, pieces of wood, pieces of metal. So the food should be free of these things. And as you know, there is always a possibility of entry of these things into your food processing chain of food processing. Food processing is not one step. You have to bring raw materials into your factory. You have to wash them. You have to process them stepwise. You have to pasteurize them. You have to sterilize them. You have to pack them. You have to put the packs in a box and send them to the market. So it's not a one-step process. It has multiple steps. And there is a risk of entering, for example, chips of wood. ලී කෑලි වැටෙන්න පුළුවන් අපේ ආහාරයට. ඒ ලී කෑලි ටින් එක කෑන් කළාට පස්සේ වැටෙන්න පුළුවන්ද? බෑනේ. raw material තියෙද්දිද වැටෙන්නේ, මෙහෙ මොකක් හරි වැඩකදීද වැටෙන්නේ? ඒක important නේද? යකඩ කුඩු. There is a grinder in the food processing chain of processing. There is a grinder, a metal grinder, which crushes food. And then there is a chance for adding pieces of metal from that grinder. එතකොට ඒක කෑන් එක හැදුවාට පස්සේද raw materials තත්ත්වෙද මෙන්න මේ පොයින්ට් එකේදීද වැදගත් වෙන්නේ. කියන්නේ යකඩ කෑල්ලක් මේ ආහාරයට වැටෙන්න ඉඩ තියෙන්නේ අඹරන තැනදී නේද? රවිදු?
Yes, sir.
Okay. There is a pasteurization step. Pasteurization. 85 degrees, 25 seconds, for example. And pasteurization fails. Bacteria grow in this food. So to test whether pasteurization works or not, at what stage you have to test it? In the raw material stage, in the pasteurization stage, after pasteurization, or after packing and send it to the market? Just after the pasteurization stage, you have to check whether pasteurization worked okay. The temperature, the heating reached up to that level. You have to check whether the temperature meters, thermometers are working properly or not. Likewise, there are many steps in the food processing event, and different hazards can enter into your food in different places. ලී කෑලි එකතු වෙන්න පුළුවන් මෙතෙන්දී. වීදුරු කෑලි එකතු වෙන්න පුළුවන් මෙතෙන්දී. පැස්චරයිස් නොවී තියෙන්න පුළුවන් මෙතෙන්දී. මිනිස්සු නාහේ ඇඟිල්ල ගහලා මේ ස්ටැෆිලෝකෝකස් අවුරියස් ඇතුල් වෙන්න ඉඩ තියෙන්නේ මෙතෙන්දී. මීට පස්සෙත් නෙවෙයි මීට කලිනුත් නෙවෙයි. ඒක දැන් තේරෙනවද? රවින්දු?
Yes, sir. Yes, sir.
So there are hazards. Hazard කියන්නේ මොකද්ද? අනුතුරුදායක නරක දේවල්. So
Yes, sir.
hazards, have we mentioned about hazards here? Glass, pieces of glass, pieces of metal, pieces of wood, soil, bacteria. These are hazards. Don't we have to know what type of hazards can enter into this food? මේක සර්පය එහෙම ඇතුල් වෙන්න පුළුවන්ද මේ ආහාරෙට? බෑනේ.
Yes.
එතකොට මෙතන possible hazards ටික identify කරගන්න ඕනේ නැද්ද අපි මුලින්ම? If you want to
Yes.
keep the food final product hazard free, don't we have to identify what the hazards are? මේකට ලී කෑලි එකතු වෙන්න පුළුවන්. මේකට රොඩු වැටෙන්න පුළුවන්. මේකට කෙස් වැටෙන්න පුළුවන්. මේකට නාහේ ඇඟිල්ල ගහලා ස්ටැෆ් අවුරියස් වැටෙන්න පුළුවන්. මේක টয়ලට් ගිහිල්ලා හරියට හොදා ගන්න නැතුව ඇවිල්ලා E. coli ඇතුල් වෙන්න පුළුවන්. පැතොජෙනික් බැක්ටීරියා ඇතුල් වෙන්න පුළුවන්. මෙන්න මේවා ටික hazards. මේකට යකඩ කෑලි ඇතුල් වෙන්න පුළුවන්. මේක ළඟ භූමි තෙල් කෑන් එකක් උඩ තියලා තියෙනවා නම් ඒක හැල්ලා භූමි තෙල් ඇතුල් වෙන්න පුළුවන්. ඒවා අයිඩෙන්ටිෆයි කර ගැනීම වැදගත් නැද්ද මේ ෆුඩ් ප්‍රොසෙසින් ප්ලාන්ට් එකේ? මේකේ මැෂින් වලට දාන්න ගෙනාවු පෙට්‍රල් කෑම වලට කලවම් වෙන්න පුළුවන්. ඒවා ටික අයිඩෙන්ටිෆයි කරගන්න ඒක වැදගත් නැද්ද? hazard identification. Identifying what the possible hazards are, is it not important?
Important, sir.
Can you give it a name? Hazard identify, hazard
Hazard.
analysis. Can you call it hazard analysis?
Yes, we can call it.
Okay, first we have to analyze the hazards. What the hazards are? ඒව දන්නේ නැතුව මේකට මේ යුක්‍රේන් කාරයෙක් ඇවිල්ලා කෑම කාලා ඇයිද කියන එක hazard එකක් නෙවෙයිනේ. ඌ ඉන්නේ යුක්‍රේනේනේ. ඒකින්ද possible hazards මේකට බල්ලෝ පණීද? කෑමට බල්ලෝ හපයි. බල්ලෝ කෑම හොරකම් කරයිද? බල්ලන්ට ඇතුල් වෙන්න බැරි නම් ඒක hazard එකක් නෙමෙයි. පුළුවන් නම් ඒකත් hazard එකක් තමයි. So identifying the hazards is called hazard analysis. Do you agree that it is an important step to keep food safe for the consumption? Ravindu?
Yes, sir. Yes, sir.
Okay. Then critical control points කියලා જાතියක් තියෙනවා. Critical points control කතාව දැන්ට අමතක කරන්නකො. Critical points from where the hazards can enter into food. දැන් බල්ලෙක් කෑම ගන්න පුළුවන්ද? මේක සීල් කළාට පස්සේ කෑන් එකකට දැම්මට පස්සේ පැක් කළාට පස්සේ පෙට්ටියකට දැම්මට පස්සේ ලොරියට පැටවුවට පස්සේ බල්ලෝ කන්න පුළුවන්ද මේ කෑම?
No, sir.
බෑනේ. ඔයා කලබල වෙනවා ළමයි කතා කරන්නේ. හරි. රයිට් කමන්නේ මං තනියෙන් කරන්නම් මේ
No, sir.
කට්ටිය උතන කතා කරනවා. හරි anyway. So different hazards can different points of the different stages of the production process, different stages of the food processing, different hazards can enter. How are we going to call these points? යකඩ කුඩු ඇතුළු වෙන්න පුළුවන් මෙතෙන්දි. හරියට පැස්චරයිස් නොවී තියෙන්න පුළුවන් මෙතෙන්දි. උෂ්ණත්වමානය කැඩුනා නම් මේක වරදින්න පුළුවන් මෙතෙන්දි. බල්ලෝ කන්න පුළුවන් මෙතෙන්දි. টয়ලට් ගිහිල්ලා අත හොදන්න නැතුව ඇවිල්ලා කන්ටැමිනේට් වෙන්න ඉඩ තියෙනවා නම් මෙන්න මෙතෙන්දි. දැන් ඊට කලින් දැන් ෆුඩ් ටික oven එකේ දාන්න කලින් টয়ලට් ගිහිල්ලා ඇවිල්ලා ඇල්ලුවත් ලොකු hazard එකක් එතන නෑනේ. එතන ඇතුළු උනත් oven එකේදී බැක්ටීරියා මැරෙයිනේ. ඊට පස්සේ ආහාර ඇල්ලුවොත් මේ ඒවට අර fecal contamination වෙන්නේ. Likewise, there are critical points in this food processing procedure. Can you understand that there are critical points? There can be වීදුරු කෑලි එකතු වෙන්න පුළුවන් මෙතෙන්දී. ගල් කෑලි එකතු වෙන්න පුළුවන් මෙතෙන්දී. යකඩ කෑලි මෙතෙන්දී. බැක්ටීරියා මෙතෙන්දී. නාහේ ඉන්න බැක්ටීරියා මෙතෙන්දී. එහෙම කියලා හිතන්න පුළුවන්ද? How can we call
Yes.
these points stages of the food processing? We can call them critical points. Can we not call them critical points? මේවා තමයි
Yes.
වැදගත් තැන්. ඒ තැන් නෙමෙයිද අපි control කරන්න ඕනේ.
Yes.
entry control කරන්න ඕනේ. entry හරි බැක්ටීරියා විනාශ කිරීම හරි enter වීම වළක්වන හරි ලී කෑලි ඇතුල් වෙන එක වළක්වන හරි ඒ points වල නේද ලී කෑලි ඇතුල් වෙන්න ඉඩ තියෙන තැනදී නේද ලී කෑලි ඇතුල් වෙන එක control කරන්න ඕනේ.
Yes.
That's why we call critical points, we call them critical control points. Why the word control is there? These are the points in which you have to control the entry of hazards. What are the hazards? That is hazard analysis. What are the points the hazard can enter into your food? Those
Critical
are critical points. Why do we call them critical control points? Because control
to
measures
control.
have to be applied in not to anywhere else, but in these critical points. So that's putting all these together, we call it hazard analysis critical control points. හරිනම් ඕක කියන්න ඕනේ analysis of hazards and identifying what are the critical control points ආගේ දිග එකක් නේ. ඒක මිනිස්සු කියනවා hazard analysis and critical control points කියලා. කියනකොටම ළමයින්ට තේරෙන්නේ නැහැ. දැන් ඒගොල්ලෝ කලබල වෙනවා. මේ මොකද්ද මේ කියලා. නම ඇහෙනකොටම අපබ්‍රංශයක් නේ නම. analysis of hazards කිව්වහම තේරෙනවද? hazard analysis කියන්නේ ඒක තමයි. Identifying critical points of entry of hazards කිව්වොත් තේරෙනවද? තේරෙනවා. ඒ වුණාට එහෙම කියන්නේ නැහනේ. Those are the points which you have to control the entry of hazards, to prevent the entry of hazards. එතකොට තේරෙනවනේ. So cutting everything short and we say hazard analysis critical control points. එච්චර තමයි කියන්නේ hazard analysis critical control points. HACCP කියලාත් කියනවා. ඕකට සෙනග බය වෙනවා මොකක්ද අප්පා මේ කියලා. Identification of hazards is important. Identifying critical points is important. Naming them critical control points is also justifiable. Now, we have to make sure that each critical point, the hazard is not allowed to enter. ලී කෑලි ඇතුල් වෙන තැන ලී කෑලි ඇතුල් වෙන්නේ නැහැ. ඒ ලී කෑලි එක්ක චෙක් කරන්න ඕනේ. එක්ක ලී කෑලි කැඩෙන්නේ නැති ලියකින් හදලා තියෙන්න ඕනේ මේ පෙට්ටිය මොකක් හරි ආහාර තියෙන එක. යකඩ කෑලි එකතු වෙන තැන යකඩ කෑලි එකතු වුණාද කියලා මොකක් හරි මැග්නට් එකකින් බලන්න ඕනේ. මැග්නට් එකකින් යකඩ කෑලි අයින් කරන්න ඕනේ. එහෙම තියෙනවනේ ඒවා තමයි control measures. අන්න ඒ control measures thermometer pasteurization කරන තැන thermometer check කරලා බලන්න එපා thermometer වැඩ කළාද කියලා. එතකොට මිනිස්සු නාහේ ඇඟිල්ල ගහනවද කියලා බලන්න face mask දාලා තියෙන්න ඕනේ. මිනිස්සු টয়ලට් ගිහිල්ලා ඇවිල්ලා කෑම අල්ලනවද බලන්න ඒක ඉට්ටු පාතක টয়ලට් එකක් තියෙන්න විදියක් නැහැ. ගිහිල්ලා එනකොට අත හොදාගෙන එක verify කරලා තියෙන්න ඕනේ. හොඳට සබන් දාලා හොඳට පිරිසිදුව එන එක. ඒ විදිහට hazards control කරන එක මහ ලොකු දෙයක් නෙවෙයිනේ depend on the hazard ඒවා control කරන්න ඕනයි කියන එක තේරෙනවද?
Yes, sir.
ඉතින් කවුරු හරි control කරනවද කියලා සැරිසරේ check කරලා බලන්න ඕනේ නැද්ද?
Yes.
That is also important. Keeping records. thermometer බැලුව මහත්තයා බැලුවා කියලා ඉඳලා හරියන්නේ නැහැ. අහවල් දිනේ අහවල් වෙලාවේ temperature එක හරියට බැලුවා calibrate කළා. thermometer එක හරියට වැඩ කරනවද කියලා known temperature එක 100 degrees වල පැහෙන වතුර භාජනයකට දාලා 100 එනවද කියලා temperature එක බැලුවා. එහෙම බලලා මේ thermometer හරියට වැඩ කරනවා කියලා දැනගත්තා. දැනගෙන ඒක ලිව්වා පොතක. ඒක record keeping වැදගත්ද? check කරන එක වාගේම check කළා බවට record එකක් keep කිරිල්ල වැදගත්ද? පවත්වා ගැනීම. Ravindu,
Yes, sir.
it is also important.
Import, yes.
ඊට පස්සේ රෙකෝඩ් කීපින් එක හරියට තියෙනවද කියලා කවුරු හරි ඇවිල්ලා සැරින් සැරේ චෙක් කරලා බලන්න එපා. ඒක වැදගත්. රෙකෝඩ් කීපින් රෙකෝඩ්ස් තියාග චෙක් කරන එක වැදගත්. ඒව ගැන රෙකෝඩ්ස් තියෙන එක වැදගත්. රෙකෝඩ්ස් තිබ්බද කියලා බලන එක වැදගත් නැද්ද?
අതെ.
බලන එකත් වැදගත්. ඊට පස්සේ ෆයිනල් ප්‍රොඩක්ට් එක ෆයිනල් ෆුඩ් ප්‍රොඩක්ට් එක ඇත්තටම ඔය කියන විදිහේ හසාඩ්ස් නැද්ද කියලා එයින් සාම්පලයක් සැරින් සැරේ අරගෙන චෙක් කරලා බලන එක වැදගත්ද?
Yes, sir. Import.
ඔන්න ඔකට තමයි HACCP කියන්නේ. Can you understand it now?
Yes, sir.
Not a big deal, isn't it?
Yes.
A logical thinking. Logical thinking. Good. So that's all about HACCP. Now you can understand it. Having in your mind the principle of the importance of each of these steps, you can understand, you can read a book that there are several steps in HACCP, and that is it. What I have explained it, nothing more, nothing less. That's all. Good manufacturing practices. One of the good manufacturing practices කියන්නේ GMP කියන්නේ ඒත් common sense. පොණ්ඩේ කඩාගෙන හිටියොත් කේස් වැටෙයි. කෑම අරගෙන ඒකට කෑ ගහ ගහ සින්දු කිව්වොත් ඒ කෙලවලින් තියෙන බැක්ටීරියා වැටෙයි ඒකට. අතින් ඇල්ලුවොත් ස්ටැෆ් අවුරියස් වැටෙයි. භාජන හොදන්න නැතුව හිටියොත් ඒවල බැක්ටීරියා ග්‍රෝ වෙලා ප්‍රශ්නයක් වෙයි. මාස් අම්බරන මැෂින් එක හරියට හේදුවේ නැත්තම් පිරිසිදු කරලා තිබ්බේ නැත්තම් ඒකේ බැක්ටීරියා වැවිලා ඊළඟට අම්බරන මස් වලට බැක්ටීරියා එයි. බල්ලෝ ඇතුල් වෙන්න දුන්න නම් උන් කෑම කයි. ඒවා ඔක්කොම ටික නැති වෙන්න වැඩ කරන එක තමයි good manufacturing practices කියන්නේ. පිරිසිදුවට වැඩ කරන එක. බැක්ටීරියා ඇතුල් වෙන්න පුළුවන්, වෙන වෙන hazards ඇතුල් වෙන්න පුළුවන්. ඒව ඇතුල් නොවෙන්න දැන් මේ බිම ඔක්කොම මාස් වතුර හලලා ඉවරලා එතන ඇවිදා හැම තැනටම contaminations ගෙනහිල්ලා ඒව කරන එක ෆෑන් දාලා කේක් රත් වෙලා කියලා ඒව නිවන එක ෆෑන් යට ඒවා නිවන් නැතුව ඉන්න එක ඒවා ඔක්කොම good manufacturing practices. ඇඟිල්ල දාලා කන එක ආහාරය හොඳ සුවඳද මේ හොඳට ලස්සනට බේක් වෙලාද කියලා බලන්න ඉඹලා බලන එක කට ළඟට ගෙනල්ලා ඒවා ඔක්කොම ඒවා නොකරන එක good manufacturing practices. හරිද? ළමයි ඔයාලා බැන්නට තරහ නැත්තම් මගෙන් අහන්න තියෙනවා නම් ප්‍රශ්නයක් අහන්න නැත්තම් මං නවත්තනවා දැන්. මමත් මිනිස්සෙන්නේ උදේ ඉඳලා මේ කියවලා ඕගොල්ලෝ ප්‍රශ්න එහෙම අහනවා නම් මාව පොඩ්ඩක් නගලා තියෙනවා නම් මම මුළු දවසෙම වුණත් ලෙක්චර්ස් කළ හැකියි. මගේ කටත් කැඩිලා ගියාට කමන්නේ මේ උගුරත් මොනවත් උනත් එහෙම වෙන්නේ නැහැ. මම මේක පුරුදුයි. ඒ වුණාට ඕගොල්ලෝ බින්දුවක්වත් සපෝට් එකක් දෙන්නේ නැතුව අපි ඉගෙන ගන්නේ නැතුවම මේකේ ඉන්නවා. බැරි වෙලාවත් ප්‍රශ්නයක් ආවත් ඔළුවට අහන්නෙම නැහැ. ඔකගෙන් හොඳ පාඩමක් උගන්වනවා මහේන්ද්‍රයට කියලා හිටියොත් ඕගොල්ලන්ට ඉතින් ඒ ලොකු අවාසියක් වෙන එක නැහැ. ටිකක් පාඩුයි. මොකද කියන්නේ? Any questions? රවින්දු ඕගොල්ලන්ගෙන් අහන්න ප්‍රශ්න තියෙනවද කියලා? මට බය වෙලාද කොහෙද දැන් ඉන්නේ?
Dear participants, if you have any more clarifications or any questions, this is the time you can ask from the resource person. Even if you can drop a message in the chat, it's okay. Anything you can ask related to microbiology. It may be not related to the topics that we discussed today. It may be related to microbiology. You can ask from the resource person.
අමන්නේ ටයිම් අරගෙන අහන්නකො ඉමින් තියෙනවනම් ප්‍රශ්නයක්.
We have finished the content, right, sir?
It looks like නේ දැන් ඔය ඇතිනේ.
Yes.
I have covered a lot of things, but if they have questions, I can end up in two different conclusions. I have the option of thinking that they couldn't understand a single thing of what I have said. Or I can guess that they must have understood more or less almost everything I said. By experience, by my experience, yes, dear, Imesha, Imasha.
Is there some standards for HACCP or GMP? International standards or whatever?
දැන් ඔය GMP කියන එක නම් ඉතින් common sense නේ. හරිද? Just HACCP, those are the standards. You have to calibrate the equipment. You have to keep recordings. You have to check whether you have kept recordings or not. You have to check the final product. Before that, you have to identify what the possible hazards are. And then you have to identify what the critical points are. Then you have to know that you have to implement control measures in these critical points, not before that, not after that. These are the critical points in which you have to implement your control measures. Those are the standards. If you follow these things and keep recordings to show that you have followed, made sure that you have calibrated your equipment, everything goes smoothly, and your final product is microbiologically, chemically, physically all right, not containing any hazards, that means you have followed the hazard analysis critical control points principle. That is the standard. Microbiology, the number of bacteria allowed to be present in this food, it depends on each different foods. Not a single E. coli, not a single salmonella. One E. coli is allowed, less than 1 in 100 milliliters. More than 1, not allowed. Those are standards. This food not contain a single salmonella. That is a standard. Single salmonella cell. Because salmonella can enter into your intestinal tract and can grow into a population and give you trouble. So your food should not contain a single salmonella. How are we going to find out your entire food is without salmonella? You can't do that practically. මුළු ඔක්කොම ආහාර ටික සල්මොනෙල්ලා වලට grow කරලා බලන්න පුළුවන්ද? බෑනේ. Not 1 grams, not 2 grams, not a small amount. You take 25 grams of food. You take 25 grams of food. That is a big sample. You take a 25 grams of food and leave it overnight. Leave it for 24 hours in a medium which favors the growth of salmonella, which is best for the growth of salmonella. එතකොට මොකද වෙන්නේ? කිසියම් ආහාරයක you take 25 grams and transfer it to a medium which is ideal for the growth of salmonella, and you come following day. So the point is, even if one salmonella cell is there in your 25 grams, a large amount, 25 grams, they would go into a huge population by following day for you to find out whether salmonella is there or not. එකෙක් දෙන්නෙක් හිටියයි කියලා ආහාරේ හොයාගන්න බෑනේ. අපි enrich කරලා බලනවා salmonella වගේ එකෙක් හිටියොත් වැරදීනේ. එකෙක් හිටියත් ඒ කෑම කන්න හොඳ නැහැ. එකෙක් ඉන්නවද කියලා බලා ගන්නවත් බෑනේ මේ ලොකු ආහාර තොගයක. ඒකෙන් 25 මිලිග්‍රෑම් අරගෙන අපි salmonella growth medium එකක දාලා enrich කරනවා. That enriching means increasing the population to a detectable size. පහුවදා එනකොට salmonella තියෙනවා ලංකාවේම මිනිස්සු ගන්නට වැඩි මෙතන හැදිලා. එතකොට අපිට salmonella ඉන්නවද කියලා identify කරන් test එකකට පටන් ගන්න පුළුවන්. If the test is positive, the food is bad. not up to the standard. So each and every bacterium, each and every food, each and every item, there are standards. Then අපි පිටරට යවනකොට කොහොබත් ඒකේ ස්ටෑන්ඩඩ් එකක් තියෙනවා. ඊකෝලයි එකෙක් දෙන්නෙක් නම් හිටියත් කමක් නැහැ කොහොබත් ඔන්න එක විතරනේ. ඊට වැඩි හිටියොත් තමයි ඒ කැමති. කන දෙයක් නෙවෙයිනේ කොහොබත් කියන්නේ. Likewise, there are different standards. And these standards come separately. Hazard analysis critical control point means how to keep hazards away from your food processing. That is an intelligent thinking and a system. You put in place a system. It is not a standard, it is a system which makes sure that your food product, the final product, is without these hazards. Have you got the point? Imasha?
Yes, sir.
ඔයා දැන් එකපාරට කලබල වෙන්න එපාදුවේ ස්ටෑන්ඩඩ් කිව්වහම මේ ඔක්කොම ඔළුවට අපිට වදයක්නේ මේ වල ඩෙෆිනිෂන්ස් මොනවද? හසබ් කියන්නේ මොකද්ද? ඕවා දෙකක් තුනක් ඔළුවට ආපුවම අපි බය වෙනවා. එහෙම බය වෙන්න එපා. Be strong. අපිට මේක තේරුම් ගන්න පුළුවන් කියලා පැත්තකට වෙලා හිතන්න කල්පනා කරලා. දැන් නිකන් තියරියෙන් හිතන්න එපා මේක ස්ටෑන්ඩඩ් එකක්ද මේක මෙතඩ් එහෙම හිතන්න එපා. ඇත්තටම මේක මොකද්ද? ඇත්තටම HACCP කියන්නේ hazards ඇතුළු නොවි ඔයාගේ ආහාර කර්මාන්ත ශාලාවේ එක එක පියවරවල් වලදී පුළුවන් ඇතුල් වෙන්න පුළුවන් විවිධාකාරයේ hazards ඇතුල් වෙන්න නොදී ආහාර ප්‍රොසෙස් එක කම්ප්ලීට් කරන විදිහනේ නේද? ආහාරයක ඉන්න පුළුවන් අහවල් බැක්ටීරියාව මීට වැඩි ඉන්න බෑ කියන එක ස්ටෑන්ඩඩ් එක. ආහාර පිරිසිදුවට හදා ගන්න ඕන කියන එක good manufacturing practices. ආහාරයට hazards ඇතුල් නොවි තියනවද කියන එක හරියටම දැන් අද මිනිහෙක් ඇවිල්ලා බලනවා. හෙට නිකා ඉන්නවා. එතකොට හෙට ෆුඩ් එක hazard එකක් ඇතුල් වෙන්න පුළුවන්. එහෙම නොවි මුළු අවුරුද්ද පුරාම food manufacturing plant එක without hazards, without allowing any hazard to enter or remain in your food processing process is what we call good, HACCP. See, there are identification of hazards, locating the critical points, knowing that you have to implement control measures in those points, to make sure regular inspection of these points, whether the control measure is successful or not, to do that, whether the equipment works okay, whether people are washing their hands, to inspect them, to keep that you have done it in records in paper or in a computer, and checking the final product, inspecting it from time to time whether the process is going according to the rule. Those are the hazard analysis and critical control points. hazard කියන්නේ ඕක තමයි. දැන් හරිද? Himasha,
Yes, sir.
are you
Thank you.
okay now? Huh?
Yes, sir.
Are you happy? Have you learned few things?
Yes, sir. Thank you.
Huh? Are you unhappy for shouting at you people?
No, no, sir.
ඔයාට තේරෙනවනේ දුවේ මේක කොච්චර අමාරුද කියලා මේ වැඩේ කරන්න මාත් මේ ටයි එකකුත් දාගෙන දාඩියක් දානවා මෙතන. උදේ ඉඳලා මෙතන ඉඳගෙන. දැන් ළමයින්ට මේක කොහොම හරි හොඳට තේරෙන්න කියලා දෙන්නම් කියලා ඉඳගත්තාම කතා නොකර ඉන්නකොට මේ කලකිරෙනවයි කියන එක තේරෙනව නේද? ඈ?
Yes,
කියන්න
sir.
ඕකේ. ඒකනේ. ඒ වුණාට ළමයි කතා කරන්නේ මොකද්ද? ඒ ළමයින්ගේ බයක් නේ හැටියක්. නේද? අනේ මන් දන්නේ මම නම් ළමෙක් නම් ඔයා වයසේදී මම අහන ප්‍රශ්න එහෙම රූඩ් වැඩක් නෙවෙයිනේ ප්‍රශ්න අහන එක ලෙක්චරර් කෙනෙක් කැමති වෙන්න ඕනේ ප්‍රශ්න අහනවා නම් හරි ඉතින් කියන්නකො තව මොනවද කතා කරන්න තියෙන්නේ හොඳයි ඒක අහපු එක IFM yes
එතකොට ISO වගේ සර්ටිෆිකේෂන් වලට බේස් වෙන්නේ HACCP වගේ ඒවා ඉන්න ඒවටත්
ISO කාර්යෝ කියලා තියෙනවා නොයෙකුත් ලැබ් එක කොහොමද පරිස්සමට තියාගන්නේ ලැබ් එකේ දැන් කැලිබ්‍රේට් කරන්න ඕනේ මීඩියා හදන්න ඕනේ මෙහෙමයි ඊකෝලයි ටෙස්ට් කරන්න ඕනේ මෙව හැම එකටම ඉන්ටර්නැෂනල් ස්ටෑන්ඩඩ්ස් ඕගනයිසේෂන් එකෙන් හදලා තියෙනවා ප්‍රොසීඩියර්ස් අන්න ඒවා ෆලෝ කරන්න ඕනේ ඒවා තමයි ප්‍රමිති ආයතනය කියන අයත් කොපි කරලා අපිට දෙන්නේ එතකොට ඒවා කලේ නැත්තම් අපිට ඉන්ටර්නැෂනල් ට්‍රේඩ් කරන්න බෑ අපේ බැක්ටීරියා අපේ ආහාරය පිටරටකින් ගන්නෙත් නෑනේ අපි ISO ස්ටෑන්ඩඩ්ස් වලට අනුව මේක කළයි කියලා කිව්වේ නැත්තම් ISO කියන එක මැෂින් වලටත් තියෙන්න පුළුවන් කම්පැනි එකේ එයාර් කන්ඩිෂනර් වලටත් තියෙන්න පුළුවන් එයාර් පියුරිෆිකේෂන් එයාර් පියුරිටි එක මනින මෙතඩ් එක අපි කියමුකෝ වාතයේ ගොඩක් බැක්ටීරියා ඉන්නවද කියලා බලන මේ සක් කරලා එයා ඒක ග්‍රෝ කරලා ඒක බැක්ටීරියා ඉන්නවද කියලා බලන ඒ ප්‍රොසෙස් එකට ISO වෙනම ස්ටෑන්ඩඩ් එකක් තියෙනවා මෙන්න මෙහෙමයි කරන්න ඕනේ මෙච්චර වොලියුම් එකක් එයා යවන්න ඕනේ හුලං මෙච්චර වොලියුම් එකක් මේක හරහා ඇදලා ගන්න ඕනේ මෙච්චර දවසක් ග්‍රෝ කරන්න ඕනේ මෙහෙම මීඩියම් එක දාන්න ඕනේ ඒ විදිහට ISO කාර්යෝ හැම එකකටම ස්ටෑන්ඩඩ් එකක් හදලා ඉවරලා ඒව විකුණනවා ක්‍රෝනා 25යි 30යි 40යි ඊට පස්සේ ස්ටෑන්ඩඩ් එක අලුත් කරනවා අලුත් කරන්නේ එක වගන්තියයි එක වාක්‍යයයි එක වචනයයි ඒ අලුත් එඩිෂන් එකේ තියෙන්නේ අර වචනේ විතරයි අනිත් ටික බලා ගන්න ආපහු අර පරණ එකක් ගන්න ඕනේ එතකොට ක්‍රෝනා 25 වෙනුවට 50ක් ගෙවන්න ඕනේ ඊට පස්සේ ඊළඟ අවුරුද්දේ ආයෙ එඩිට් කරපුවම තව 25ක් ගෙවන්න ඕනේ ඉතින් ආහාර කර්මාන්තයේ ඉන්න අය ISO ස්ටෑන්ඩඩ්ස් ඉන්න අය ඕක ඉතින් මේ ඕවා ස්ටෑන්ඩඩ්ස් අරගෙන කාට හරි ආයතනයකට දීලා ISO ස්ටෑන්ඩඩ්ස් වලට තියෙනවද කියලා සර්ටිෆයි කරගෙන තමයි ISO තියෙනවයි කියලා ලේබල් එක ගහන්නේ. ඉතින් ISO කියලා දැක්කහම ඉතින් හොඳයි. ඉතින් ඒක සේෆ් නේ. ඒකේ උපකරණ සේෆ්. උපකරණ හරියට වැඩ කරනවා. උපකරණ වලට ISO අරන් නම් උපකරණ වැඩ කරනවා. ඒකේ UV ස්ටෙරිලයිසේෂන් ඒකට ISO අරන් නම් UV හරියට වැඩ කරනවා කියලා දැනගත්ත හැකියි. දැන් මිරිස් අඹරන ආයතනයක ISO ගහලා තියෙන්නේ මිරිස් කුඩු වලටද පැකේජින් මැටීරියල් වලටද මිරිස් හදන මිරිස් මෝලේ එන්වයරන්මන්ට් එකටද කියලා දන්නේ නැතුව හරියට ISO කිව්ව ගමන් අපිට පිළිගන්නත් බෑ. ISO කියලා නම්බර් එකක් තියෙනවා. ඒ නම්බර් එක අපි හොයලා බලපුවහම මිරිස් කුඩු වල ප්‍රමිතියටද එහෙම නැත්නම් මිරිස් මෝලේ වාතාශ්‍රය පිළිබඳ ප්‍රමිතියකටද හරියට වෙන්ටිලේට් වෙනවද කියන ප්‍රමිතියටද මිරිස් අඹරන මිනිසුන්ගේ නාහෙට මිරිස් කුඩු යන්නේ නැහැ කියලා මේක් ෂුවර් කරලා ඒ ප්‍රමිතියටද ඒකට දාන මාස්ක් එකේ ප්‍රමිතියද අපි දන්නේ නැහැ. අපි ඒක හරියට නම්බර් එකත් එක්ක බලන්න ඕනේ මොකක්ද මේ ප්‍රමිතිය කියලා. මොකක් හරි එකක ISO කියලා දැම්මට වැඩක් මේ හරියට අපිට තේරෙන්නේ නැහැ සාමාන්‍ය මහ ජනතාවට. ISO නම්බර් එක මොකක්ද බලලා ඒ නම්බර් එක අරගෙන තියෙන්නේ මේකේ වාහනවල දුමටද මේ ෆුඩ් වලටද එහෙම කියලා බලන්න ඕනේ. හරි. අහපු එකකටත් නෙවෙයි මම උත්තර දුන්නේ චුට්ටක් වැඩියෙන්. ඒක තේරුණා නේද? ඒ උනාට ISO කියන්නේ මොකක්ද කියලා?
Yes sir. Thank you.
ඔයාලා මොකද කරන්නේ? ඉමාෂා මොකද කරන්නේ?
මම මේ කැලණියම undergraduate කෙනෙක් මේ applied chemistry third year.
Applied chemistry APCH.
Yes sir.
APCH. මාව දැකලා තියෙනවද?
Yes sir.
හොඳද මං?
හොඳයි.
ළමයි මොකද කියන්නේ මං ගැන?
එහෙම ලොකුවට අහලා නැහැ මේ අපිටත් මයික්‍රෝබයොලොජි මොඩියුල්ස් තිබ්බා. ඒවා අපිට ඉගන්නේ වෙන ලෙක්චරර් කෙනෙක්.
හරි. අහන්නකො මයික්‍රෝබයොලොජි කරන ළමයි ඉන්නද යාළුවෝ?
ඉන්න සර්. ස්පෙෂල්
අහන්නකො මං ගැන මහේන්ද්‍ර සර් කියන්නේ කොහොමද කියලා අහන්නකො මූසලේද්ද දැන් ඕගොල්ලන්ට බැන්නාම ඕගොල්ලෝ හිතයිනේ හෙන්න නරක මිනිහෙක් කියලා මං ඒක අඩු කරගන්න මේ අහන්නේ අනික් ළමින්ගෙන් අහන්නකො ඇත්තටම මූසලේද්ද කියලා බෑ
අහන්න.
අහන්න කියලා කියන්නේ බය නැතුව නරක දෙයක් කියන්නේ නැහැ කියලා දැනගෙනනේ. හරි.
හරි සර්.
IPCH නේ.
APCH.
APCH. IFM 0028 mute unmute කරගෙන මම ඉන්නේ කතා කළේ නැහැ වචනයක්වත්. කමන්නේ. එහෙනම් මම නවත්තන්නද දැන් රවින්දු how is it? Do you think I missed anything?
We covered everything, I guess.
So okay then.
So if you have any more questions, I guess we can conclude the session, sir.
Okay. Okay.
Yes, so we have come to the end of the today's session. So dear participants, I would like to take a moment to express our sincere gratitude to Dr. Mahendra Gunawardana for these great explanations and for clear and very insightful explanations. So this is our third batch. So thank you all for your participation and thank you all once again. So we hope to see you in our future sessions and have a great day. Thank you so much, sir, for your time and everything. Thank you so much.
You're welcome. So then see you
We'll
sometime
conclude the
again.
session.
Yeah, please. Bye for now.
Okay. Bye.